Good morning, ladies and gentlemen.
Good morning, Judy.
Hi, Graham. Sorry, that was ladies and gentlemen and Graham. Okay, no problem. So, a very warm welcome to you all. Some of you have come from very far-flung reaches of the planet to be here today. And all of you have had your scientific and or your social paths have crossed with Marcello's, at least for some time in the last 4 decades or so. This meeting we designed to bring together some of Marcello's closest colleagues so they can showcase their scientific achievements, which may have been enhanced or may even have occurred in spite of their interactions with Marcello over the years. The occasion was planned, as some of you have heard already, not because Marcello has made any decision to retire from scientific life, but instead so the rest of us could get together before we all retire and leave him forging on into the scientific future. Indeed, this occasion brings me out of retirement to once again try and keep a group of speakers in line and in time. In particular, it gives me one last opportunity to try and keep Marcello to 15 minutes and less than 100 slides. So nothing gives me greater pleasure than to try one more time. To set the scene, I'd like to give a brief overview of Marcello's scientific career for those who don't know it intimately. And include in that, weave in some messages from some absent friends and my own personal tribute. I'd just like to mention again, for those of you who knew Peter Steele was a very fabric of the scientific life of many people in this room, even those who didn't work at Flinders, who visited Flinders and took advantage of the fantastic facilities that Peter had an integral role in setting up as the first laboratory manager in the School of Medicine. Peter unfortunately died 2 days ago and we remember him very fondly. So Marcello completed a medical degree with a research thesis at the University of Torino, and after compulsory military service he lectured in the Department of Anatomy in Turin. He came to Australia in 1970, lured by the larger-than-life Geoffrey Burnstock, who assembled an amazing group of researchers in the zoology department at Melbourne University. He really brought together an enormous range of techniques, and not only that, but an enormous a group of researchers with completely diverse backgrounds, and bringing them all together in the same place, that is the zoology department at Melbourne Uni in the 1970s. Geoffrey couldn't be with us today, but he sent his greetings to Marcello. Really sorry not to be with you for the celebration, but my warmest wishes for a great occasion. I much enjoyed our work together and friendship over the years, and he's included a few photo memories. This one we've seen already. I've worked out that this must have been 1974, and here's a very young Marcello. Now this, Jeff obviously has some doubts whether this is Marcello. Perhaps Marcello might confirm or deny this. I'm not actually sure whether that's formal swimwear attire, Marcello, or... and is that a very young Andrea in the front row there? In the front with Marcello and Jeff and Naomi. A few years ago, Marcello. How many is that? Marcello and his sparring partner, Max Bennett. And this was the famous book that Elspeth referred to last night, Bernstock and Costa, alphabetical obviously, Marcello. But this book they published had a great influence on neuroscience. Yes? Would you prefer that they didn't refer to their colleagues, Elspeth? So it wasn't in the early 1970s in zoology that Marcello teamed up with John Finesse, and they started their prolific collaboration that continued after they both moved to Flinders as foundation members of the School of Medicine. Now, it was in 1975 that I first met Marcello. I was a fresh young honours student in zoology, student of Graham Campbell's. He's taught me everything I know. Well, all the polite things anyway. And both Graham and Marcello held up the long-standing zoology tradition, or maybe they created the long-standing zoology tradition of a procession over to the coffee lounge at Melbourne University several times a day. I remember one particular time proceeding across the quadrangle with Marcello and Graham and a nubile young technician that accompanied Marcello, where I witnessed for the first time of several Marcello giving instructions to the staff behind the counter on how to make a good coffee. That's stuck in my mind since 1975. Indeed, at the end of 1975, I almost moved to Flinders myself. I was offered, I think, probably one of the first PhD scholarships by the School of Medicine. I was tossing up whether to come and do a PhD with Marcello and with John to the great environment they were just on the verge of just starting to set up at Flinders. But there were one or two reasons that kept me in Melbourne for a few more years, and it wasn't until 1984 that I came to Flinders with Ian to work in Marcello and John's laboratory, and they themselves, dare I say, had really, I shouldn't say follow the Bernstock tradition, but in the best of Bernstock tradition, amassed an amazing group of researchers from around the world, had set up a multidisciplinary approach to the neuroscience, which has really been the strength of work in the lab at that time. Now, the reason I have particular personal gratitude to Marcello for my arrival at Flinders was that Marcello actually sat my National Heart Foundation fellowship interview while I was in the States. He went along and was me. I did get the fellowship, so he must have done something right. So thank you, Marcello. I may not have been here without that. Now, the period of the 80s was an amazing one, with Marcello and John applying these approaches. And I really feel very lucky to have been at Flinders at that time. It was an amazingly exciting time where we were discovering new things almost every day. They were fantastic times, going in the little dark room down the corridor and just seeing lights glowing in the dark, seeing molecules in places people didn't know before. It was amazing. Marcello has, throughout my career, helped me along the way. He's helped me to see the path of righteousness. He has maintained his cheerful approach to the world, helped me put everything in perspective, and at the same time helping me develop as an independent scientist. So Marcello, I do owe you a debt of gratitude for that. Now, at this time, all this amazing histochemistry was happening at Flinders. There were one or two labs around the world who were really on a similar path. One of these was the Karolinska Institute in Stockholm, where Thomas Herkfeld and Jan Lundberg were really tackling similar problems to what was happening in Flinders. Again, Thomas sends his apologies. He couldn't be here today, but he sends these greetings. Dear Marcello, sorry for not being able to participate in the MarcelFest 08, but I send my warmest regards from Karolinska and Stockholm to you, who I've admired and whose splendid work I've followed for decades, almost as long as I can remember. Yours, as always, Thomas. P.S. I cannot guarantee that all Swedes will hoist the flag, and I haven't spoken to the royal family yet, but I'm sure had they known you, they would join me in saluting you and your achievements. So just briefly continuing with Marcello's career, he has stayed working at Flinders since 1975 when he arrived, making a huge impact on neuroscience locally in Australia and around the world. He's played a key role in setting up the Centre for Neuroscience at Flinders and in establishing and making strong the Australian Neuroscience Society. He was convener of the Centre for Neuroscience and is now a life member of the Centre for Neuroscience and has been president of the Australian Neuroscience Society. He was elected to the Australian Academy of Science in 1988. He's also more recently played a key role in establishing the South Australian Neuroscience Institute, bringing together neuroscientists from the 3 South Australian universities and other neuroscientists around Adelaide. He's received numerous awards, local, national and international, for his scientific achievements and for his passion and skill at communicating science to the broader community. In fact, it was only 2 weeks ago that Marcello and Ian Gibbins were awarded Unsung Heroes of Science awards for their communication of science in South Australia. Marcello's developed many successful collaborations locally and around the world, and I'm not going to tell you any more about that because I'm going to hand that over to the very people who are at the core of these collaborations. So, without further ado, I will ask John Finesse to come up and say his words after I just get his presentation. There we go, John. Space or just arrows?
Okay. Well, it's a great privilege to lead off this talk today. I've deliberately not tried to develop a formal talk because I just want to say things.
Can I just interrupt? Yes? If you're going to stand, can you use the radio mic so I can
okay.
say it's recorded?
Who can't hear me?
No, it's recorded.
Oh.
Oh, raise my phone. No, sorry. Let's not talk about it.
Oh, okay. I'm a wanderer. I'm sorry about that.
Yeah.
Okay. I'll do my best. I'll try. When I walk over there, just shoot me back here. Okay. Anyway, it is a great privilege to lead off today. And as I said, I've put some things together, but I haven't planned the exact talk. I just want to talk about what sort of comes up. Now, it has been a wonderful time that I had working with Marcello for very many years, and I'll show you a slide in a moment that documents that if you like. I learned more from Marcello than I think I've learned from anybody else about science, how to approach science, life, how to approach life, and it was an absolutely wonderful time. Eventually, I migrated to Melbourne for various reasons, and I'm there now, but I keep assiduously following what's happened here, and the productivity and the imagination of Marcello and his group has been absolutely fantastic. This is a little picture here. I don't have a pointer, but anyway, you can see it. So this is Marcello in Melbourne in 1971 when we were working together there. His cheeky grin. See, he's not always looking glum, Marcello. This is more like him, I think. And of course, here it is in Adelaide in 2008. Now, we started working together at that time. Marcello came to Melbourne with some radical theories about the organisation of the nervous system, and we set about trying to... Oh, there we are. Oh gosh, this is slower than my finger. I'm trying to go backwards. There it is. Oh, what have I done? I'm losing control. So we published together for over 20 years. So that's the first paper we published in 1971 on these cells that Marcello had actually discovered, which were pretty unusual at the time. And then we published all the way through the early 90s together. That's our most recent paper together. Maybe there'll be some other things in the future, who knows. But in between those two, this space here is filled with a hell of a lot of stuff. And we ranged over lots of different techniques because I guess maybe I'm not nearly as clever at ideas, but I love techniques. So I'm mucking around with some sort of stuff in the lab, building something or doing something different. And we ended up with an enormous range of histochemical and physiological and electrophysiological and even combined technologies that were a tremendous amount of fun. And Marcello was always full of ideas. There was always a new challenge to do something more interesting and different and that nobody else had done before. So that was a tremendously interesting time and as I say, a lot of fun. Now I'm going to see if I can move one. Got it. And this is a paper we published together in those early days, which became a citation classic, as they say. And we spent a lot of time thinking about where you should go in science. And of course, this session is meant to be about where you should go. And this paper, which we wrote together, sort of set out a little framework. It sort of said, this is what we ought to do. And the fundamental thing was, we ought to try and correlate all the information about these neurons. We ought to understand how their physiology fits with their anatomy, with their chemistry, with their places in circuits, and put it all together. And it was a lot of blueprint. And we worked on that problem for the next 10 years to try and do that and to, as Marcello once said, unravel the circuitry. And it was interesting that we spent time thinking about approaches, not also just jumping into things. Of course, Marcello, as you know, is a good thinker and a good talker, so we had long conversations about where we should go and what we should do, often on into the night. But it was really productive to do that. I'm trying again. Oh, there it is. And after I went to Melbourne, Marcello continued on this sort of work and really sorted out the classifications of neurons in this rather complex system, which has been incredibly important because that's advanced to studies in human and parallels in other animals. And this is a classic paper that Marcello published, which deals with what we call the chemical coding. And I've put up next to that the paper where this idea was first in the literature. We looked at a fairly minor question, but we saw in that answer to that question a principle that was much broader. And so this paper was the first one that talked about this idea of chemical coding. And we developed a sort of theory around this, and the theory is a very simple one, that every class of neurons must fit into the circuit in a different way. So every class of neurons should have some sort of chemical signature, which we call a chemical coding. And that hypothesis has carried on now. And I even, in the last month, I picked up a paper, I can't remember who it was by, somebody working on the brain or some other minor part of the nervous system. And in it was said, and the chemical coding hypothesis was written into this thing. And that's where it came from, this little study. But sometimes you've got to see what the broad implications of a small study was. And Marcello was very good at that, of seeing that, okay, we've studied a little tiny problem, but that problem illustrates a biological principle. And that was the way Marcello thought, and I think it was very, very productive. And it's amazing that, I don't know when this was published, 1984. Anyway, it's still talked about. And Marcello then, much later, 2002, I was in Melbourne at this time, edited this book with Simon, and this book that they organised shows the transformation of a field that when Marcello started working on this 20 years before, or 30 years, I can't remember, a long time anyway, it was so poorly understood, it was almost like, you know, walking in the dark. But he singularly sorted out the system, and this book, published now 6 years ago, defines the way in which this nervous system works, and it's to great credit of Marcello and also younger college at that time, like Simon, that they sorted all this problem out. So I'm just going to run very quickly through a few things because I don't want to talk for too long. So this is the sort of periods of decades. So in the 1970s, when Marcello was first in Australia, he really set out to solve some problems of circuitry and set up the basis for doing that. And I was privileged to be involved in that. And then really, once the basis for solving the problem was sorted out, and we published that citation classic in 1980, then the next decade really was to, in Marcello's word, unravel the circuits. And he succeeded in doing that. And that provides a basis that is now in all the textbooks. So it's good to think that you start off on a problem that is obscure and you end up with it being proliferating the textbooks. And that's what happened. And then in the 1990s, a lot of the detailed information was sorted out, and you saw the front page of that book that Marcello and Simon published. And then he moved along to a new stage, I say, in the 2000s, which is a reaching out stage, where communicating science to people, getting out into the community, broadening the base for understanding science. So it's really a progression from starting off dealing with minutiae in a way to broadening out to deal with generalities. So it's been a fantastic career. So I was asked to say, where do we go from here? Well, it's impossible to say where we go from here because we don't know. But we can think about a few things. So we're talking about the enteric nervous system, the system that controls the digestive system. And really the foundation's now laid. We know the circuitry, we know how it's organised, we know the major neurons, we know the transmitters. But we know a lot of those things for the brain too. But how the hell does it generate the behaviour it generates? So the next stages are going, I think, to be in computational neuroscience. That's putting this together using computational methods to understand the system. And computational neuroscience, now with the power of computing and the details we can get out in experimental terms, is going to be a new part of the future. This is also in the brain too, but this system, because of its simplicity, will be the first mammalian system that's really tractable. And you'll know that the famous psychiatrist Eric Kandel got a Nobel Prize. Now he's a psychiatrist. What did he get his Nobel Prize for? Working on molluscs. Because the difficulty of understanding integration in the nervous system is profound. He got a Nobel Prize for working on molluscs. Now, if you're going to translate that to mammals, you have to look at some slightly simplified system in mammals, and this is an ideal system. So I think this is where one of the things will go. The other thing is pathology. Now, there are a lot of enteric pathologies causing different disease states and disorders, but there are no good ways of understanding them because we didn't know the circuitry. We didn't know what the underlying basis was. We do know it now. So now to develop animal models of enteric pathologies and to translate that to human is another thing for the future. a big hope is in live imaging. We're now able to image the brain down to single neuron level. If we can also image in live animals and people, what's happening in this nervous system with great temporal and spatial resolution, and the techniques are coming, then we'll have a wonderful window. We won't any longer deal with abstractions, we'll deal with the real thing. So I think that's another part of the future. And of course, tissue engineering, the replacement of neurons using stem cells, using genetic modifications. This is another frontier. So Marcello's laid the basis. He says, this is how the normal system's organised. Now we can go and find out how it computes and how it's involved in pathology. So I think that I can say that Marcello's work has laid a fantastic foundation. It's set the scene, and now it's up to you younger people, while us old farts sort of watch on, to take the next step and to use this sort of information to solve real problems that are basic to understanding how the nervous system works and understanding disease and helping people. So, thank you, Marcello, for your fantastic contributions over the years, for your friendship, for your guidance, and for everything you've done for all of us.
So next on the list, all the way from Manchester in the UK, we have Alan North.
Thank you very much for asking me to come and speak on discoveries in neuroscience. What next? If I knew the answer, would I tell you? Huh? Mind you, I was entertained when John suggested that all you young people out there will be doing things in the future, because I don't actually see an awful lot of young people in the audience. But maybe you were referring to me, John. I'm not going to spend too much time on reminiscing. There is always a tendency on occasions like this to sort of delve into nostalgia. But I thought I might just remind you, so I sat there thinking about it myself, that in 1974 I turned down an offer from Laura Geffen to come and work in Flinders. I remember it was 73, so it was about that time. In fact, I'd already looked into how I would get there. I was going to drive down to Sri Lanka, and then I checked out the boat timetables that I could ship my car across to Perth or Fremantle or somewhere. And then, you know, as often happens in these occasions, then I got a phone call from another part of the world, so I finished up in the United States. I mean, I did eventually get to Flinders and had a very productive 6 or 8 weeks working with John and Marcello, and I think that was in about 1983.
First in the literature, we looked at a fairly minor question, but we saw in that answer to that question a principle that was much broader. And so this paper was the first one that talked about this idea of chemical coding. And we developed a sort of theory around this, and the theory is a very simple one, that every class of neurons must fit into the circuit in a different way. So every class of neurons should have some sort of chemical signature, which we call a chemical coding. And that hypothesis has carried on now. I even, in the last month, I picked up a paper, I can't remember who it was by, somebody working on the brain or some other minor part of the nervous system, and in it was, and the chemical coding hypothesis was written into this thing, and that's where it came from, this little study. But sometimes you've got to see what the broad implications of a small study was, and Marcello was very good at that, of seeing that, okay, we've studied a little tiny problem, but that problem illustrates a biological principle. And that was the way Marcello thought, and I think it was very, very productive. And it's amazing that, I don't know when this was published, 1984. Anyway, it's still talked about. And Marcello then, much later, 2002, I was in Melbourne at this time, edited this book with Simon. And this book that they organised shows the transformation of a field that when Marcello started working on this 20 years before, or 30 years, I can't remember, a long time anyway, it was so poorly understood. It was almost like, you know, walking in the dark. But he singularly sorted out the system, and this book, published now 6 years ago, defines the way in which this nervous system works, and it's to great credit of Marcello and also younger college at that time, like Simon, that they sorted all this problem out. So I'm just going to run very quickly through a few things because I don't want to talk for too long. So this is the sort of periods of decades. So in the 1970s, when Marcello was first in Australia, he really set out to solve some problems of circuitry and set up the basis for doing that. And I was privileged to be involved in that. And then really, once the basis for solving the problem was sorted out, and we published that citation classic in 1980, then the next decade really was to, in Marcello's word, unravel the circuits. And he succeeded in doing that. And that provides a basis that is now in all the textbooks. So it's good to think that you start off on a problem that is obscure and you end up with it being proliferating the textbooks. And that's what happened. And then in the 1990s, a lot of the detailed information was sorted out, and you saw the front page of that book that Marcello and Simon published. And then he moved along to a new stage, I say, in the 2000s, which is a reaching out stage, where communicating science to people, getting out into the community, broadening the base for understanding science. So it's really a progression from starting off dealing with minutiae in a way to broadening out to deal with generalities. So it's been a fantastic career. So I was asked to say, where do we go from here? Well, it's impossible to say where we go from here because we don't know. But we can think about a few things. So we're talking about the enteric nervous system, the system that controls the digestive system. And really the foundation's now laid. We know the circuitry, we know how it's organised, we know the major neurons, we know the transmitters. But we know a lot of those things for the brain too. But how the hell does it generate the behaviour it generates? So the next stages are going, I think, to be in computational neuroscience. So putting this together using computational methods to understand the system. And computational neuroscience, now with the power of computing and the details we can get out in experimental terms, is going to be a new part of the future. This is also in the brain too, but this system, because of its simplicity, will be the first mammalian system that's really tractable. And you'll know that the famous psychiatrist Eric Kandel got a Nobel Prize. Now he's a psychiatrist. What did he get his Nobel Prize for? Working on molluscs. Because the difficulty of understanding integration in the nervous system is profound. He got a Nobel Prize for working on molluscs. Now, if you're going to translate that to mammals, you have to look at some slightly simplified system in mammals, and this is an ideal system. So I think this is where one of the things will go. The other thing is pathology. Now, there are a lot of enteric pathologies causing different disease states and disorders, but there are no good ways of understanding because we didn't know the circuitry. We didn't know what the underlying basis was. We do know it now. So now to develop animal models of enteric pathologies and to translate that to human is another thing for the future. A big hope is in live imaging. We're now able to image the brain down to single neuron level. If we can also image in live animals and people what's happening in this nervous system with great temporal and spatial resolution, and the techniques are coming, then we'll have a wonderful window. We won't any longer deal with abstractions, we'll deal with the real thing. So I think that's another part of the future. And of course, tissue engineering, the replacement of neurons using stem cells, using genetic modifications. This is another frontier. So Marcello's laid the basis. He says, this is how the normal system's organised. Now we can go and find out how it computes and how it's involved in pathology. So I think that I can say that Marcello's work has laid a fantastic foundation. It's set the scene, and now it's up to you younger people, while us old farts sort of watch on, to take the next step and to use this sort of information to solve real problems that are basic to understanding how the nervous system works and understanding disease and helping people. So, thank you, Marcello, for your fantastic contributions over the years, for your friendship, for your guidance, and for everything you've done for all of us.
So next on the list, all the way from Manchester in the UK, we have Alan North.
Thank you very much for asking me to come and speak on discoveries in neuroscience. What next? If I knew the answer, would I tell you? Huh? Mind you, I was entertained when John suggested that all you young people out there will be doing things in the future, because I don't actually see an awful lot of young people in the audience. But maybe you were referring to me, John. I'm not going to spend too much time on reminiscing. There is always a tendency on occasions like this to sort of delve into nostalgia. But I thought I might just remind you, so I sat there thinking about it myself, that in 1974 I turned down an offer from Laura Geffen to come and work in Flinders. I remember it was 73, so it was about that time. In fact, I'd already looked into how I would get there. I was going to drive down to Sri Lanka, and then I checked out the boat timetables that I could ship my car across to Perth or Fremantle or somewhere. And then, as often happens in these occasions, then I got a phone call from another part of the world, so I finished up in the United States. I did eventually get to Flinders and had a very productive 6 or 8 weeks working with John and Marcello, and I think that was in about 1983. So let's not spend too much time in nostalgia and just get straight to the bottom line. And basically, neuroscience will be dictated by the same factors that dictate the rest of science. And that is the rise in computational power. And a couple of recent events I just thought I'd tell you about, which kind of bring this home to you. And these events were just a couple of months ago. Manchester, some of you may know, is the home of the computer. And I'll say a couple of words about a birthday party that we had 2 months ago. And then the other thing that I think it's worth mentioning is that a couple of months ago, the scientists at IBM declared that they'd now produced a machine that would operate at this sort of magic figure of 1 petaflop. Now, for those of you that are not kind of in the business, a flop is a floating point operation per second. And peta is 1,000 times tera, which is 1,000 times giga, which is 1,000 times mega. So we're talking about 10 to the 15 floating point operations per second. This was quite an achievement. There'd been a big sort of international competition to get machines running at this speed. I also mentioned to you that we had a bit of a birthday party. In fact, some of the people that had created the baby in Manchester, which first ran in June 1948, came back to the university. And Steve Ferber, who is a professor of computer science at the university, gave us a lecture in which he described the 50 billion-fold increase in computer power since 1948. So it's a 50 billion increase in operational speed of computers. The baby weighed 1 ton and occupied an entire room. And it was the first example of a machine carrying out a calculation from a stored instruction, or a program, as you like. As Steve Ferber pointed out, had there been a similar increase in technology around the internal combustion engine over the period of 60 years, then the entire vehicle population of the United Kingdom would run for 1 year on 1 litre of petrol. Or put another way around, the world reserves of petrol would be enough to keep us busy, keep us in fuel for the predicted lifetime of the solar system. So we have presided over these 60 years over fantastic increase in computer power, and I don't see much sign of that slowing down. Now, you might think, well, what's this got to do with neuroscience? Some of you will remember the days of oscilloscopes, photographing the faces of oscilloscopes and then sitting in dark rooms and projecting the images and measuring them up. Pretty hard to imagine that we were doing those kind of strange things. Yeah? Likewise, in the imaging field in general, the technology which we now have available to help us is quite remarkable. John's alluded to some of it. Positron emission tomography has now got resolution in human brain, getting down to well below millimeter resolution, 300 micron resolution. MR imaging is huge. The ability to make mice which express proteins that emit light or proteins which fluoresce in light in certain subsets of neurons over very defined periods of time is quite a remarkable advance. In networks, we've already said something about. I'm not sure whether the GI tract will be the first place where neuronal networks really drive us forward. But I think if any of you know Dennis Noble's work on the heart, then you cannot fail but to be impressed by what you can produce from a computer which so closely replicates the function of an entire organ. And that's getting down to 20 or 30 channels in each cell and 100 or 200 different proteins in the metabolic pathways of each cell. All the cells put together functioning like a pretty good heart, at least good enough for most of the pharmaceutical industry to use it to test their drugs and see whether or not they produce anti-dysrhythmic side effects and so on. Similar developments are occurring for kidney, for lung, and so on, and pretty soon they will be occurring for pretty large parts of the nervous system. AI, of course, is going extremely quickly, and I suppose in 25 years' time, then there will be no need for humans to even devise experiments because they will all be devised for you. So what the future then of the artists from Ramon y Cajal up to Marcello Costa? Many of you have not seen this slide before. Some of you saw the finished version last night. But this is Marcello out in the open air recreating that version de novo. Very little need for this kind of artistry in the future, I suspect, although there will be some diehards, such as Marcello and myself, that still rather enjoy doing this kind of thing. So what's going to be the consequence of this rise in computational power? Two things I'd really like to dwell on for a moment. And the first of these is what you might call the demise of serendipity. It's happening already, and we can all think of great examples of it. Serendip is an interesting word. Serendip is the old name for Sri Lanka, and there were some princes of Sri Lanka that actually introduced the word into our lexicon. And the original example that was given a couple of hundred years ago was the example of someone walking down a lane and noticing that the grass on one side of the pathway was shorter than the grass on the other side of the pathway. And they concluded from this observation that the lane had previously been traveled by a donkey that was blind in one eye. So this is the origin, and gives you in many ways a kind of a good feel for what is commonly meant nowadays by serendipity. You can all think of your own examples. I've come up with a few. I mean, barbiturates are a good one because it's about 100 years old. In neuropharmacology, phenobarbitons had been around as a hypnotic for a number of years. And then just about 100 years ago, a German physician who was taking care of epileptic patients was having his sleep disturbed. These were in the days when the physicians used to sleep above the wards. And he got tired of having his sleep disturbed by the epileptic patients who were fitting during the night. So he thought, well, I'll just sedate them a little bit with some phenobarb. And this was his accidental discovery that phenobarbital is also a very good anti-epileptic drug. It's still in use as a primary anti-epileptic drug 100 years later. Many of you will know with Bob Furchgott's remarkable discovery of nitric oxide. he accidentally, or someone in his lab accidentally left a piece of endothelium attached to a blood vessel. And when they put an acetylcholine, instead of contracting the blood vessel, lo and behold, it relaxed the blood vessel. And then he eventually noticed that the relaxation was stronger when the room was dark than when the room was light, because the transmitter was destroyed or broken down much more rapidly in the light time. Albert Hofmann and LSD is another example. In 1938, he discovered the psychedelic actions of LSD by tasting it himself. And this led to a whole new area of neuropharmacology. One of my own best examples from my sort of personal experience is actually came from a conversation. This was when I was at the Volume Institute, and it came from a conversation in the hallway between myself and a virologist. This is someone who was working on an ecotropic retrovirus in a mouse. What did I know about virology? Precious little. But he'd asked myself and actually Mike Kavanagh, who was working with me, if we could express this particular cDNA in an oocyte, because he'd heard that we were expressing things in oocytes. And I said, yeah, sure, we can express it in an oocyte. Why do you want us to do that? He said, well, I want to make some protein so that we can raise antibodies against it. And I asked him what kind of molecule this was that he wanted us to express. So he said, well, this is just a receptor for a virus. And so we expressed it duly, and then Mike Kavanagh thought, well, this is weird. This is a membrane protein. Maybe this is some kind of channel or transporter. This was at the time when only one transporter had been cloned. It was 1990. The only transporter that was known about was the glucose transporter, the sugar transporter that Ernie Wright had cloned. And so Mike Kavanagh figured perhaps this was some other kind of protein. And after a few experiments, testing on the oocyte what caused currents to flow, he deduced that in fact this retrovirus receptor was a basic amino acid transporter, the first basic amino acid transporter to have its structure determined. The entire experiment, which of course led to the development of a big field in terms of transporter physiology, the entire experiment arose from a hallway conversation between a virologist and a biophysicist. I suspect that this kind of serendipity, fun though it has been, is pretty much on its way out. The second thing then that I think the rising computational power is going to bring about is what you might call the decline of inductive reasoning, or basically the decline of hypothesis-driven science. And that largely is a result of the massive amounts of information currently available to us. And I'll give you just one or two examples of that. So the huge amount of information that is now available, particularly in our field as a result of genomics, but also as a direct result of informatics, means that the kind of old-fashioned way of doing science, if you like, which Karl Popper called the inductive hypothesis-driven approach, is largely being overtaken by deductive methods, which are of massively high throughput. I don't think there will be a complete revolution, because the output of all these large high-throughput deductive methods is in fact to promote new hypotheses which can then be tested. But the methods of testing the hypotheses very often, because of the volume of the approach, very often finish up giving answers that we never possibly could have predicted. So hypotheses are essentially looking for keys under lamplights. In order to make a hypothesis, you need to be able to see something, whereas these high-throughput deductive approaches don't have any lamplights at all. And some of the examples just that you could use to illustrate that, one of them came out in Nature a few months ago, and it's perhaps one of the best ones. This arose from 3 parallel studies, each of which used whole genome associations. Whole genome associations are now becoming increasingly common because of the possibility of sequencing entire genomes. So each of these studies used a couple of thousand people, anywhere between 2,000 and 6,000 people, And there were different populations of people. One was in Iceland, one was in Europe, so they just all happened to be published at the same time, even though they were done independently. These several thousand people all had their genome sequenced, and then those genome sequences were compared with the likelihood of these individuals having got lung cancer. Now, you could have expected any kind of result from this. You could have found that no genes had mutations which were associated with lung cancer. In fact, all these three studies found the same answer, and that was that there was a mutation, or actually a whole series of mutations, in the same region of nicotinic receptor subunits. There were two nicotinic receptor subunits involved which were mutated. One was the alpha-4, the other was the beta-3 subunit. So three independent studies all saying that if you have this mutation in either your alpha-4 or your beta-3 nicotinic receptor subunit, you are more likely, much more likely, to have lung cancer. It's quite a remarkable observation. Who would have thought that nicotine was acting on nicotinic receptors to produce lung cancer, yeah? Everyone says it's the tar and the other additives in the cigarettes. And who would have thought that it's nicotinic receptors because they're on neurons, yeah? Not in the lung. So this is an example of this massively high throughput, deductive kind of science now driving us back to new hypotheses. It's very likely the nicotinic receptors concerned are on lung macrophages or on lung epithelial cells. And there's something about that which makes you more likely to get lung cancer. Although a couple of these papers also suggested that because this particular nicotinic receptor is also found on brain dopamine neurons, this might actually give you a susceptibility to start smoking in the first place, because these are the reward neurons, the dopamine pathway of the brain. So I think there's still controversy around whether or not these mutations make it more likely that you will smoke cigarettes, or whether they simply make it more likely that you will get cancer if you smoke cigarettes. As soon as whole genome association studies start picking off one disease after another, I think we can expect many more sort of major surprises of this kind. So Karl Popper himself noted that much of the future lays beyond our predictive capabilities. It's a kind of profound thing that philosophers such as Popper came out with. I actually think that Yogi Berra had got it a little bit more better summed up when he said the future ain't what it used to be. There'll still be time for a good bit of animated argument, as is obviously going on in this particular example. But I rather suspect that Flinders neuroscience will have gone a different direction from the one which is depicted in my final slide in 1983. I suspect that many of these experimental physiologists here will have already crawled inside the waste bin, and the lid will have been closed on them, but only the guys sitting on the ground there doing the thinking will be surviving. Thanks very much.
I forgot to mention before that the coffee and tea at the back there are available any time for you to get up and get a cup. You might need it before the next speaker. Who's come all the way from Bedford Park. My pleasure to ask Bill Blessing to come and say a few words. This is going to take a few minutes to load, so if you just hit there when it's finished. Okay?
Well, I'm a lucky man because my hospital in Sydney was invaded by John Chalmers, and he said, come down to Adelaide. And not only that, when I got there, he said, go up there and learn something and make sure you tell me everything. And up there was to John and Marcello, and I had the advantage that I wasn't actually their student, and also I was a physician, so I could pretend that I was more important if I needed to. So they yelled at their other poor PhD students, and I listened. I thought, God, I was just about to do that experiment down in John's lab. I won't do that one. it's not clear in my mind as to which was John and which was Marcello in those days, because they both had the same enthusiasm, the same drive, and to me the same critical. They were the first people I met that I realised that, in a sense, ordinary people could be scientists. I used to think that you had to be a mathematical expert and have all the equipment up on the wall, but John and Marcello and Laurie Geffen and Robert Rush too, we were lucky because we realised that, well, we didn't realise it, but they were just good scientists, and I was lucky to be there. The first thing they did for me was give me my own tiny little bit of the brain using their method, which actually, to be honest, I hate the gut. I don't like the gut. I hate the gut. But they gave me their method and they said, have a go at the brain. And luckily, there were little nerve cells in the brain, here down in the bottom of the medulla, and I was the first person ever to see this. I was lucky I got to the microscope before John and before Marcello, and I was able to show it to John Chalmers. And this was the first 10 years or so of my research was based on working out how these particular little nerve cells, which synthesise noradrenaline, how they actually worked. I even, years later, made a model of one of these cells, which is not widely published, because I couldn't convince people that it wasn't an onion weed that I dug up from my own garden, and indeed it was, but my God. It does look good. Because it wasn't that I didn't like the gut, but over the years, the concept of the autonomic nervous system gradually started to make me think that, where the hell was it? And what system was it out there that was doing things? And gradually, and here was the first time in some ways that Marcello came with me in this idea, and I think I made a contribution with this, that I realised that there wasn't really the brain and the periphery. There wasn't a system out there. Maybe there is a little bit in the gut. But for the rest of the body, if you deprive it in mammals, if you deprive it of the brain, it doesn't do much. And in a sense, I was lucky because working on the brain, I felt that I wasn't stuck with one bit of science. I could move wherever I wanted. Now, I'm not going to go into more detail in this, but it actually changed my way of thinking about how you should do science. And for the next 10 minutes or so, I'm going to be like the ancient mariner, because with the help of my colleague Yo-Yo Utskar, who is a colleague in my lab, I think we've discovered something that I think is new. And I think it's serendipitous, and we didn't have too much fucking computational power either. Well, maybe we had a bit. So I'm going to skip to and I'll come back to that in a minute. What we measured the way animals make heat when you put them in the cold. And up the top here is a trace where in rats they have this special tissue, the same tissue that bears and so on have when they hibernate, the brown babies have, brown adipose tissue, and they actually make heat. So when you pick up the rat and you put the rat, just hold it in your hand and then put it in the fridge, its body temperature in the middle trace there, it stays constant. But a lot's happening because the animals got this sort of feed forward, and feed forward is the opposite of what we were taught as students. We got taught that your body temperature dropped, that triggered off all the different responses, and so there were corrective mechanisms that sent you back to the set point. And indeed, half of physiology that we're taught is like that. But that's because it was done in anaesthetised animals. In the real situation, the thing sees ahead. So the moment you step out into the snow, the brain knows that you're cold. So look, it does. It constricts all the blood to the tail straight away before you get cold. And in the rat, it makes heat. Now, making heat was what, and measuring this brown adipose tissue was what led to what I think is a very interesting thing. What we did was just leave the animal and not do anything. And this trace here goes for a whole day. And we measured the temperature of the brown adipose tissue. It was already known in the middle trace that if you don't interfere with the system, your body temperature goes up and down a bit. But there is a literature on it, but it's really not much. And who knows why that happens. It could have been, maybe the earth shook a bit, who knows. So people didn't worry. But what we discovered was that this was associated with increases in the brown adipose tissue. And the brain tells the brown adipose tissue to make heat. Now, why would it do that? It was funny, because if there's a set point and you're trying for homeostatic regulation of body temperature, then if the temperature's going up, which it is, why would the brain tell the brown adipose tissue to make heat? What's more, when I went to some of my colleagues and said, let's have a look at your 24-hour records, and they'd never looked at the record from an individual animal because the computer, thank God for computational power, it gives you a mean of 10 at a time. But I said, no, I just want to look at one of your animals, and look in the middle. This is for you, John Chalmers. In the middle there, that's the arterial pressure of a rat. And the next one down is its heart rate, and the heart rate's going up 100 beats a minute. Every hour, the rat's heart rate, or every 90 minutes, its heart rate and its blood pressure, and its body temperature, and its brown adipose tissue, and down the bottom, it's moving around. It goes up like this, in this funny way. And over the last 6 months, I've thought about this a lot, and I saw this picture. And this is to me, it was a very revealing thing that I think has given us a new idea. This little guy's a hamster, a special one. It lives out in Siberia or somewhere, but I think ordinary hamsters are the same. They have a kind of hibernation every day. Hibernation is when the animal goes down and let me just summarise it. There's two sorts of animals. There's ones called ectotherms, like lizards, and what they do is let the outside world heat them up, the sun or so. Then there's mammals and birds heat themselves up, so that a lizard will be 20 degrees if it's 20 degrees outside, but surprisingly they can get in the sun and bask and so on and warm themselves up, but they don't use their own energy to warm themselves up. On the other hand, mammals, somewhere inside, it's not actually known exactly so far where the heat's coming from, but we keep our temperature at 37. Now the point is that even at normal environmental temperatures, we use between 5 and 10 times more energy than a lizard warmed up at the same temperature. And a lot of scientists have thought about that, because why, if it's 5 or 10 times more energy, you need 5 or 10 times more food. And actually, even though we got too much food in our particular environment, in the world as a whole, people are still starving, and the food has always been a limiting factor. And there are strategies that animals have taken to protect themselves. What an animal does, if it doesn't have enough food, it changes from an endotherm to an ectotherm. And that's what I've realised hibernation is. Well, I'm not the first to realise that. Here's the animal's actual metabolic rate. The one that's got the sawtooth and then drops down really low and comes up again, that's the amount of oxygen that the animal's using every minute. And you can see that in the daytime, when the animal goes into torpor, its metabolic rate drops to something like 15% of its normal one. So it uses only 15% of its fuel. At the same time, its body temperature drops. What caught my attention, because of what we'd already discovered with the brown adipose tissue, is that during the time when it's awake and running around, it goes up and down just like our brown adipose tissue does. So that if the big fall in metabolism in the middle is to save energy, maybe then the little falls during the day also save energy. And that particular up and down like that 24 hours a day, that's called an ultradian or ultradian, I don't know. I didn't know the word a year ago, an ultradian rhythm. And there's a literature on ultradian rhythms, and excitingly, the first ultradian rhythm discovered was REM sleep in humans. REM sleep in humans, the more you study it and the more you learn about it, the more amazing it is. And without going on for too long, I'm like the Agent Marin, I can go all day on this. The man who discovered REM sleep, I got his picture right at the end. He's not in the rubbish bin. Well, he did die, but he was 92. He said that it happens during the day as well. It's not just that at night it happens. In animals, it's part of, he called it the basic rest activity cycle. And in the 70s and 80s, there were many, many papers in humans where they tried to sort of measure your IQ to see if it fluctuated every 100 minutes. The problem was they didn't measure the right things. And in humans, half of our evolution is to deny evolution. We don't do the things that we've got the sort of programs in there for because we've evolved the flexibility not to do them. And that's part of the magic of being a mammal. But in fact, in a human, carefully measured, their metabolic rate fluctuates like this, day and night. That's measured and published, but the significance not really realised. Why then would you need to have this fluctuation? Well, why does an animal need to be 37 or 38 degrees rather than 20 degrees? It's not really completely known, but most of the theorists say the brain works better when it's hotter. Like your car, it has to warm up properly so that if it's cold, you can't think straight. And if it's 37, you can't think as well as if it's 38. Who would have thought? Actually, it's published, you go through the literature, there's plenty of papers, but they didn't really think about it. Who would have thought your brain goes up and down in its temperature 1 degree or 0.8 of a degree? This is a rat. It's not proven in humans. But this is REM sleep. It's already known that your brain temperature goes up and down in REM sleep. But the idea that it's happening in the day when you're awake, that's a new idea. What's happening here is that you have to have your brain running strong at a higher temperature in order to get E equals mc squared. Now, I said this to John Finesse and he said, oh yeah, well, what about birds? Do they get E equals mc squared? And I was temporarily knew that he got me. However, birds do get E equals mc squared. There's a lot of intelligent birds. How many intelligent fish are there? How many fish can do tricks? There's schools of fish, but they can't do tricks. They can't do tricks, but birds can, you see. So in some ways we're affiliated with birds. Someone said dolphins. A bit of biology there. So what's happening? For some reason, you might say, well, how do we know it's true? Well, we know it's true now because what we did was measure the rat's EEG. And this is a hard slide because it's got the Z dimension. Coming out this way is the colour. And when it's white, the EEG is saying it's the EEG of an animal that is aware of the environment, an EEG of an animal that's concentrating on things. Also, the EEG of an animal in REM sleep. because if it happens to be asleep, or in the day they sleep, this will be their EEG. That might be a bit complex. The brown adipose tissue temperature, measured at the same time, is the red trace. And when you do cross-correlation between the percentage of this awakeness in the EEG and the brown adipose tissue for a 24-hour period, it's about 0.4, which is quite high. But you have to slide it first because the EEG is about 5 minutes ahead of the brown adipose tissue. So that means that in the brain, in there is some circuitry somehow, totally not understood at present, that generates this rhythm that does go day and night. And it divides our life into these little packets of activity. Very obvious in animals, not so obvious in humans, but it's a fundamental sort of thing. Now, have I got another minute? No, I finished, sorry. Okay. The last thing is, I think it's related to mental illness, but it's lucky I haven't got a minute because when I told my daughter my theory, she said, "Dad," she said, "don't say that in public. And if you do, wear a helmet and dark glasses." So, so thank you very much.
Thanks, Bill. It's now my pleasure to introduce Ian Gibbins.
Okay, so it's a real pleasure to be up here this morning talking about some of the things that have gone on between Marcello and the rest of the world in science at Flinders over the last 25 years since I've been there. It's actually also good following on from the other talks as well. And many of the points that the previous people have made will turn up in various ways in this little one. And it's also good following on from Bill too, because the part of the intellectual environment of Flinders, which has been so important, I think, for us all, is not just Marcello and John Finesse, but Bill Blessing's been a critical part of that too, and that's really important. So, I suppose if people around the place think, you know, what does Gibbons do? You generally think of being autonomic nervous system sort of stuff, and a lab's called the Autonomic Neurotransmission Lab, and indeed for much of the time I've been doing research, that's what I've worked on. But I'm actually going to, for this morning, just talk about something else that we've been interested in right from when I first came to Flinders, and sort of dropped out for a while, and now it's back again. And it's to do with the sensory end of things. And in doing this, I'll go through and actually illustrate a couple of the things which Alan said about computational stuff, because that's been part of this whole story. And then we'll finish up at a point where the future might be going, at least for Marcello. I don't know about everybody else. So we've heard about this. Someone made the comment it was hard to tell who was doing the work. Was it Costa or Finesse? So when we heard about this guy over the border, you know, this Costa and Finesse, and sometimes it wasn't always clear who was who, and they sort of merged into each other sometimes. And when we first came to the lab, I came as a Heart Foundation Research Fellow. I was actually in the Department of Physiology then, with Laurie Geffen as my first boss. And John and Marcello had little rooms just off the lab rather than having big proper offices. And that was really fantastic because they were always there listening in and just be able to pop around the corner when something interesting was happening. And it's part of the reason people in my lab, that's why I've got I do most of the work just sitting in the lab rather than in my office. That's something I learnt from both of you. So when I came to Flinders, my project was actually not on autonomic nerves, it was on sensory nerves. And the neuropeptides had just been discovered, and John talked about the chemical coding stuff. I'll come back to that as well. And at the end of my PhD with Graham Campbell, we've been it was a fantastic transition time when all of a sudden people realised there's this huge array of other transmitters out there. One or two at a time, but the work from Thomas Herkfeldt's lab in particular opened up. And then we realised from in Melbourne that over in Adelaide, John and Marcello had also in on all of this as well. And so we started figuring out, getting interested in all these neuropeptides. And one of the things that was absolutely clear right from the early stages was the conceptual frame of thinking, more than one transmitter. Well, maybe these nerves have more than one way of sending a message. But also it was tied in with the technical innovations and things. I'm just going to focus on the microscopy side of things for now. And I was really lucky when we ended up in Melbourne to have sort of almost inherited just about a whole microscopy unit that had been set up by the people we've heard about already. And over in Adelaide it was progressing on as well. So innovative microscopy has been one of the things which has been a theme the whole time we've been there. And also innovative phototechnology, which we're illustrating here. Looks like a standard double label picture everybody has all the time now, but the people from the lab will know that this was done under a dissecting microscope with real old-fashioned bits of film, and you try and get the exposures right, and you sit there with your scissors and this funny little black masking tape stuff, and you only had one shot at doing it, because if you cut the wrong bit off, you could never get it back. If you put your fingers on it, you would never be able to project it. And I've still got a cupboard full of these things, because I can't bear to throw them out. And the other thing that we did was, that was happening was the development of multiple labelling of immunofluorescence. Now again, Flynn has had a pioneer. This is Bob Rush sitting over there, one of the first people to do immunofluorescence in the nervous system. And about the time I arrived, it was clear that we really needed to get more labels, more ways of doing things. And we spent a lot of time mucking around with technology, getting all this stuff to work. And a lot of us were involved in this. But this is some interesting technology as well, because this is one of the first sort of diagrammatic slides made on a computer that I did. There's an old program called Draw Perfect, which is still one of the easiest drawing programs ever made. Except the only way of getting a slide was to photograph it off the screen, because we didn't have a slide screen. So I used to set my camera up at night and photograph the screen, just like Alan said about the electrophysiology. And here are two more ways that you could that making slides as well. We've got the notion of chemical coding, and I'll come back to that in a moment. But we need to talk about these slides too, because two other ways we made slides was diagrams like this. You'd set them up and draw them up on big sheets of proper drafting paper with rotary pens, which always blocked. You'd photograph them, get the negative, and then under the microscope, you get these special pens. There was one brand only which worked, and you'd colour them by hand. And you'd do 3 or 4 versions of them because they'd run and all the rest of it. And the other one, again, is from an even earlier computer version of making slides that I used, which was actually a statistics program, SAS, where I had to program in every pixel for where everything was going to be. The letters were already formed, but you had to tell it whereabouts on the page they had to be and what colour it was. It seemed to be worth it at the time, I don't know why. But it's only, anyway. Because we knew the technology was coming. But the important thing is this chemical coding, and was already mentioned, John's already talked about that. And by working on the sensory nerves, the nerves which respond to pain and inflammation, or noxious stimuli, inflammation, tissue damage and things like that, we sort of realised that there are these amazing patterns, and different types of nerves with different combinations of peptides were going to different places. We had no idea what all these different peptides did or what it all meant, but it certainly gave us a huge tool for identifying what was what and where they went. And Judy mentioned before, there really was a stage there where every time we went to the microscope, we saw something no one had ever seen before. Probably for about a year there, in about 1986 or something around about there. It was really fantastic. As new antibodies came in, and John and Marcello, between them, had contacts all around the world. And I still think between us, between the various labs, John in Melbourne and Marcello and Simon and us, we've probably got probably the biggest collection of antibodies for neuroscience anywhere, I suspect.
One text says we've got enough for 12,000 years.
Yeah, well, I'm sure that's true. The other thing that's worth pointing out too with this labelling tech is we realised at the time there's huge commercial benefit could be gained here in this area. And none of the Australian companies we spoke to, remember the Solinus and others, were actually interested at all, and to their disadvantage in the end, because it turned out to be a big industry doing all this sort of stuff. So what does it all mean? Judy and I were then started to diverge more from from the enteric nervous system stuff. Judy going out into the vasculature, and I started looking at sympathetic and parasympathetic ganglia, but still with the notion of this chemical coding. And Judy spent an enormous amount of time doing just the sort of things that we talked about before, combining the histochemistry, the neurochemistry with the physiology. And there were some extraordinary things came out, which, this is still 15 to 20 years ago, we realised that you could have neurons which could have the same time release something which makes the target tissue speed up and slow down at the same time. And we took that in our stride, and I remember having fantastic conversations around the lab with all of us, arguing about what this all means. And the famous day in the penny dropped with Judy, that we actually had excitatory inhibitory transmitters in the same nerves. Now, the brain people are just starting to catch up on that, and you still get papers in Nature by saying that it's some bit of the brain. But we've known it and taken it for granted for nearly 20 years now out in the periphery. So the brain's not all it's cracked up to be. So whoever said that was a good idea. Now here's another, this is where we're getting just to closing the loop. When we're doing all this sensory nerve stuff back in sort of late 80s, it was sort of obvious by thought experiments, though there wasn't very much information out there to help you decide, that it couldn't be as simple as what everybody was saying. There had to be more to it than just these peptides. There was something wrong. There was a mismatch in there between the physiology and the pharmacology and the neurochemistry and what people were talking about. And I found, it actually blew me, I wasn't quite sure what I was going to talk about, and I found this slide and I was going back looking for the old ones. And this is back 1987 or 8 or something like that, and I must have made it for a talk somewhere. And what we're talking about here is the idea that you've got two populations of nerves, these ones and these ones, both responding to potentially painful stimuli, noxious stimuli. And the idea we had then that the peptides are only doing some of the job, and that there's another population of nerves which may have been doing some other component of the job. Maybe the sharp pain compared to the dull pain. Maybe the acute pain compared to the chronic pain. Maybe the stuff which is affected by analgesics compared to the pain which isn't. It was clear that you couldn't have one population of nerves, even for all the chemical coding, that could do all those things. And that idea just sat there, and it's got and over the years, it's still been an issue, and Marcello and I have talked about a lot in recent years about we've been missing something. And the clues started to come from entirely by accident. And about 7 or 8 years ago, one of the big holy grails, I suppose, for light microscopy is to try and find some way of identifying synapses, the connections between neurons, without having to go to electron microscopy. And I was trained electron microscopist, so I know how much work it is. It's a lot of work, it's a lot of fun, you get huge information for it, but geez, it takes a long time. And you only get to look at a very, very small bit of tissue. And so in the early 90s, people have started to understand that the protein chemistry of synaptic transmission and a lot of the peptides involved, proteins involved have been identified, and good antibodies are available. I thought, beauty, we'll use these, just tell you where synapses are. No, they don't. What it did tell us was that you can actually have synapses which seem to lack all of the protein machinery that you'd normally expect to see in a synapse. And we still don't know what that really means, but there's huge variation in how things are organised. And as part of doing that, Judy and I realised that these sensory nerves that we'd barely looked at for 15 years didn't have these proteins either. And I thought, what's going on here? How could they work? How could they possibly transmit anything if they didn't have the sorts of machinery that people normally expect to be involved in releasing transmitters like glutamate and acetylcholine and even noradrenaline and things like that. So part of the rest of the clue here came from the discoveries in, I don't know what it was, about 2001, something like that, of the proteins which are involved in transporting glutamate the excitatory transmitter in the central nervous system, into the vesicles before it's released. And 3 types of vesicular glutamate transporters are identified, numbers 1, 2 and 3. Number 3 is not much interest to us. Number 1 is mostly used by all sorts of different nerves for fast transmission. But glutamate transporter 2 seem to be identifying a population of sensory nerves responding to damage in the tissue that we hadn't actually seen before. And if you look at mouse spinal cord, you see this is just up the superficial part of the spinal cord. There's beautiful layers of the different types of nerves. There's the blue ones, the red ones, the green ones, and the red ones. Again, it's all we need to know for now. The blue ones are one set of pain fibres, presumably. The green ones are IB4 ones, people know about that, probably responding to acute pain. And in between, we've got these glutamate ones. But there's no glutamate stuff in the peptides. They couldn't. This goes back to the same thing. How could they possibly work using glutamate as a transmitter? So how can you find out what's going on? How can you find out which glutamate, which ones are which? The clue here came from, again, the collaboration at Flinders with Simon. Simon Brooks is talking next. And he and his lab had worked out how to put dye into nerves and let the dye go into the ends of the nerves. You put it here and let's see where it goes in a really sophisticated, smart way. And we took that technique and modified it a little bit about a year ago now, so we could see into the spinal cord. And the picture on the black and white one here is the dye being applied to the nerves going into the spinal cord. You can see where they go and see just about all of them. And then you can colour them up with all the various markers that we normally use. And what it turns out, this is sort of results hot off the press. When I've been figured this out just in the last few weeks, really, hit the critical mass of data, is that there really are 2 sets of nerves sensory nerves going to almost the same place. It really looks like that this idea we had nearly 20 years ago, which everybody forgotten about, including us, is probably is part of the explanation of what's going on. And so now, using the new computing technology, this is just a single image from a 3D reconstruction where we can go into 3D space and do all sorts of image mathematics on the images and selectively pick out stuff. So the colours here mean things to do where things are in 3D space. And it's actually for the cognoscenti, there's 2 populations of nociceptors converging into lamina 1. The blue ones, here they are, let's do another reconstruction of them. The purple ones and the yellow ones. The yellow ones are the peptide ones, and the purple ones are the ones which contain the amino acids. So the amino acids, there's one set of nerves contain the amino acids, another set contain the peptides. They converge onto little groups of cells in the dorsal horn, side by side to each other, in these little microdomains. We've got no idea yet where the purple nerves are coming from in the body. My guess is they're coming from the same place as the other ones. And so what's this mean? It means that you've got within the neural circuit sensing damage to your tissues or pain or something like that, there's multiple ways of recording that information. Indeed, I just said that. We also know, the question let's throw up is that, well, how many sensors have you got? Here's a picture I took in my kitchen of a whole bunch of kitchen ingredients. And over the last 10 years, it's been extraordinary developments in molecular biology combined with biophysics, identifying the channels which are activated by chilies and mint and ginger, cloves, pepper, mustard, cinnamon. They all activate highly specific receptor channel molecules on the ends of different types of nerve cells. And none of the accounting yet for how all these sensory nerves project into the spinal cord has been able to pull that information together. So, in a way, I think this field of these visceral sensory nerves, these nociceptive fibres, it's at the point where John described beautifully where the enteric nervous system was in 1980, when you did your review, saying this is what we've got to do. We've got all the bits and pieces, now we've got to start pulling it together to make sense. I think only now we're starting to get the bits and pieces together to make sense of the input side of pain, of what leads to pain. And I think there's a long way to go. I think we're still way off the mark still. So then, let's bring it back to Marcello. And one of the things Marcello's been emphasising a lot in the last few years is how these nerves, the bits which are part of one nervous system. That's an important point that Bill's made. I've embraced that concept of Bill. It's this one nervous system, but nevertheless there is all this stuff out there. A phrase we've used sometimes, it's the secret nerves, the nerves you don't have much control over. There's stuff going on that you aren't really aware of. They're really important. That's the stuff which is keeping you alive. Those sort of nerves conk out and you're in real trouble. But the interesting thing is, what happens if those nerves aren't being activated? Well, they're just sitting there doing nothing. Well, you feel really good. And it's a fair chance that some of these nerves we've been talking about, the whole time you're alive, if you're really lucky, will never ever be activated. So I hope I never get a pancreatic cancer, a gallstone, a kidney stone, a really bad urinary infection, a broken leg, or any of those other things which activate these nerves. So I haven't done any of those things yet. And if I get hit by a car on the way here today, they never would have been activated the whole way out. And that's a fairly amazing concept. There's a whole chunk of your nervous system there, perhaps not doing anything until you really, really need it, until it really matters. And so an idea of Marcello's, we've talked about a lot, is this sense of well-being, the sense of feeling good, is maybe a default state, unless all these bad things happen to you. That's not a bad idea, I reckon. So I'll leave it there.
Thanks, Ian. Just to finish off this first session before a short break, Simon Brooks has already leapt onto the stage and he can now work his own presentation. Thank you, Simon.
Well, thanks very much. The first thing I'd like to say is a big thank you to Ian and particularly to Judy for organising this. It wouldn't have happened without them, and it's been absolutely fantastic. It's really nice to be able to give a tribute to Marcello. It's a bit of a challenge. There's never enough time for these things. To try to compress 20 years of science that I've shared with Marcello into just a few minutes is almost impossible. So this isn't really a Festschrift for Marcello, it's more of a short shrift, I think. Anyway, what I'd like to tell you about is details. I do this unapologetically because I think the beauty in science comes from the accumulation of what I shall refer to as those pithy little details. I think that science will be transformed, as Alan said, by the systematic approaches that are allowed by informatics, by big-scale computing, by the Genome Project. But I would also disagree with him because I suspect that in 20 years' time, it'll still be the smart people who get the best results, who make the big discoveries, and it'll be the smart people who communicate with each other, who don't work in isolation, who will be at the top of science. And I think this is something that we learned yesterday, that we don't work as islands in science. We're actually...
Come back to that as well. And at the end of my PhD with Graham Campbell, we've been it was a fantastic transition time when all of a sudden people realised there's this huge array of other transmitters out there. One or two at a time, but the work from Thomas Herkfeldt's lab in particular opened up. And then we realised from in Melbourne that over in Adelaide, John and Marcello are also in on all of this as well. And so we started figuring out, getting interested in all these neuropeptides. And one of the things that was absolutely clear right from the early stages was the conceptual frame of thinking, more than one transmitter. Well, maybe these nerves have more than one way of sending a message. But also it was tied in with the technical innovations and things. So I'm just going to focus on the microscopy side of things for now. And I was really lucky when we ended up in Melbourne to have sort of almost inherited just about a whole microscopy unit that had been set up by the people we've heard about already. And over in Adelaide it was progressing on as well. So innovative microscopy has been one of the things which has been a theme the whole time we've been there. And also innovative phototechnology, which we're illustrating here. Looks like a standard double label picture everybody has all the time now. that the people from the lab will know that this was done under a dissecting microscope with real old-fashioned bits of film, and you try and get the exposures right, and you sit there with your scissors and this funny little black masking tape stuff, and you only had one shot at doing it, because if you cut the wrong bit off, you could never get it back. If you put your fingers on it, you would never be able to project it. And I've still got a cupboard full of these things, because I can't bear to throw them out. And the other thing that we did was, that was happening was the development of multiple labelling immunofluorescence. Now again, Flynn has had a pioneer. This is Bob Rusher sitting over there, one of the first people to do immunofluorescence in the nervous system. And about the time I arrived, it was clear that we really needed to get more and more labels, more ways of doing things. And we spent a lot of time mucking around with technology, getting all this stuff to work. And a lot of us were involved in this. But this is some interesting technology as well, because this is one of the first sort of diagrammatic slides made on a computer that I did. There's an old program called Draw Perfect, which is still one of the easiest drawing programs ever made. Except the only way of getting a slide was to photograph it off the screen, because we didn't have a slide screener. So I used to set my camera up at night and photograph the screen, just like Alan said about the electrophysiology. And here are two more ways that you could that making slides as well. We've got the notion of chemical coding, and I'll come back to that in a moment. But we need to talk about these slides too, because two other ways we made slides was diagrams like this. You'd set them up and draw them up on big sheets of proper drafting paper with rotary pens, which always blocked. You'd photograph them, get the negative, and then under the microscope, you get these special pens. There was one brand only which worked, and you'd colour them by hand. And you'd do 3 or 4 versions of them because they'd run and all the rest of it. And the other one, again, is from an even earlier computer version of making slides that I used, which was actually a statistics program, SAS, where I had to program in every pixel for where everything was going to be. The letters already formed, but you had to tell them whereabouts on the page they had to be and what colour it was. It seemed to be worth it at the time, I don't know why. But it's only, anyway. Because we knew the technology was coming. But the important thing is chemical coding, and was already mentioned, John's already talked about that. And by working on the sensory nerves, the nerves which respond to pain and inflammation, or noxious stimuli, inflammation, tissue damage and things like that, we sort of realised that there were these amazing patterns, and different types of nerves with different combinations of peptides were going to different places. We had no idea what all these different peptides did or what it all meant, but it certainly gave us a huge tool for identifying what was what and where they went. And Judy mentioned before, there really was a stage there where every time we went to the microscope, we saw something no one had ever seen before. Probably for about a year there, in about 1986 or something around about there. It was really fantastic. As new antibodies came in, and John and Marcello, between them, had contacts all around the world. And I still think between us, between the various labs, John in Melbourne and Marcello and Simon and us, we've probably got probably the biggest collection of antibodies for neuroscience anywhere, I suspect.
One of my texts says we've got enough for 12,000 years.
Yeah, well, I'm sure that's true. The other thing that's worth pointing out too with this labelling tech is we realised at the time there's huge commercial benefit could be gained here in this area. And none of the Australian companies we spoke to, remember the Solenis and others, were actually interested at all, and to their disadvantage in the end, because it turned out to be a big industry doing all this sort of stuff. So what does it all mean? Judy and I started to diverge more from from the enteric nervous system stuff. Judy going out into the vasculature, and I started looking at sympathetic and parasympathetic ganglia, but still with the notion of this chemical coding. And Judy spent an enormous amount of time doing just the sort of things that we talked about before, combining the histochemistry, the neurochemistry with the physiology. And there were some extraordinary things came out, which, this is still 15 to 20 years ago, we realised that you could have neurons which could have the same time release something which makes the target tissue speed up and slow down at the same time. And we took that in a stride, and I remember having fantastic conversations around the lab with all of us, arguing about what this all means. And the famous day in the penny drop with Judy, that we actually had excitatory inhibitory transmitters in the same nerves. Now, the brain people are just starting to catch up on that, and you still get papers in Nature by saying that it's some bit of the brain. But we've known it and taken it for granted for nearly 20 years now out in the periphery. So the brain's not all it's cracked up to be. So whoever said that was a good idea. Now here's another, this is where we're getting just to closing the loop. When we're doing all this sensory nerve stuff back in sort of late 80s, it was sort of obvious by thought experiments, though there wasn't very much information out there to help you decide, that it couldn't be as simple as what everybody was saying. There had to be more to it than just these peptides. There was something wrong. There was a mismatch in there between the physiology and the pharmacology and the neurochemistry and what people were talking about. And I found, actually blew me, I wasn't quite sure what I was going to talk about, and I found this slide and I was going back looking for the old ones. And this is back 1987 or 8 or something like that, and I must have made it for a talk somewhere. And what we're talking about here is the idea that you've got two populations of nerves, these ones and these ones, both responding to potentially painful stimuli, noxious stimuli. And the idea we had then that the peptides are only doing some of the job, and that there's another population of nerves which may have been doing some other component of the job. Maybe the sharp pain compared to the dull pain. Maybe the acute pain compared to the chronic pain. Maybe the stuff which is affected by analgesics compared to the pain which isn't. It was clear that you couldn't have one population of nerves, even for all the chemical coding, that could do all those things. And that idea's just sat there, and it's got over the years, it's still been an issue, and Marcello and I have talked about a lot in recent years about we've been missing something. And the clues started to come from entirely by accident. And about 7 or 8 years ago, one of the big holy grails, I suppose, for light microscopy is to try and find some way of identifying synapses, the connections between neurons, without having to go to electron microscopy. And I was a trained electron microscopist, so I know how much work it is. It's a lot of work, it's a lot of fun, you get huge information from it, but geez, it takes a long time. And you only get to look at a very, very small bit of tissue. And so in the early 90s, people had started to understand that the protein chemistry of synaptic transmission and a lot of the peptides proteins involved have been identified and good antibodies are available. I thought, beauty, we'll use these, just tell you where synapses are. No, they don't. What it did tell us was that you can actually have synapses which seem to lack all of the protein machinery that you'd normally expect to see in a synapse. And we still don't know what that really means, but there's huge variation in how things are organised. Now, as part of a big community who interact, and the number of people who've come from all over the country and all around the world to this meeting pays tribute to that. So I would refer to this as the intellectual ecosystem. And today what I'd like to do is to talk just about the little corner of Marcello's ecosystem that I've been occupying for a while now, and tell you about some of the little details. The other theme that I'd like to bring out is to talk about some stuff to do with scientific honesty. Because we all like to think of ourselves as honest, but we actually have to work very hard at that, to be objective about things. And I would say that there is one part of scientific process in which we are often fundamentally dishonest, which is when we write up our papers, we pretend that what we actually present in our paper is actually what we intended to do when we set out. It's not true in most cases. And I would like to give you a few little pithy details from my own history with Marcello about some of these changes in direction and some of the serendipity which I think will survive in spite of all the large-scale computing and stuff like that. So I did a PhD working on this guy many, many years ago at Bristol University, and at the end of that I needed to change fields because there wasn't any future in this. So I applied for a job to work on something that I'd never heard of called the enteric nervous system. I was working with a guy called David Wingate in London, and I went to the interview on the train up from Bristol and read up in my physiology textbook about the enteric nervous system. Now, there were actually 2 sentences in that entire textbook about the enteric nervous system, so I finished that within half an hour and got to the interview, and I actually got the job. So I worked in London, got through the job interview, and worked with David Wingate and Wendy Hewitt in the Royal London Hospital in Whitechapel, £60 on the Monopoly board and grossly overpriced at that. And
After about 3 years, I'd done a few projects which had worked reasonably well, one of which was to record from human enteric neurons for the first time, diafill them. But I realised that my project and my career really wasn't going as fast as I wanted. And in fact, my career was really taking off something like this. And this was because there was nobody in the lab who could really supervise me very well. They didn't understand those pithy little details. So I decided that I needed to move on. And this is where the start of this network actually happens. Because I wrote 2 letters to people that I'd met in the previous year. I wrote a letter to Alan North and to John Furness, saying what I'd done, that I wanted to come to their labs and to learn some new techniques and to work with them. And what I really wanted to do was to understand how a productive lab really works. Well, I got a reply back from John Furness saying that he didn't have any positions for me, but that he had passed on my letter to Marcello Costa. What I didn't realise was this was about the time that the labs were being split. And Marcello wrote to me a few weeks later saying that he did have a position for me to come out to Australia. So we moved from London to Adelaide. Helen and I got married, and then a couple of months when we'd sorted out all the visas, we came out to Adelaide and met Marcello. Well, actually, we didn't meet Marcello. He wrote to us that he's going to meet us at the airport. And when we got there, we got off the plane, came into Adelaide Airport, no Marcello. Now, there was a bit of a problem here, because I'd never met Marcello and had no idea what he looked like. Marcello had never met me and also had no idea what I looked like. And of course, Marcello had rushed from an experiment and hadn't actually taken the time or thought in advance to have one of those little signs that said Brooks on it to meet me at the airport. So consequently, it took half an hour for everybody to disappear off the flight. And eventually, there were just 3 people upright in the airport, 2 of us with big suitcases standing there. And Marcello then, using his philosophical powers of deduction, figured out. And then he drove us down to Flinders in his beaten-up old 1962 Holden station wagon. He drove us down South Road, past the car scrapyards and the tattoo parlours and the strip joints, and Helen and I looked at each other and said, what the hell have we done here? Well, we got to Flinders, and I resisted the temptation to go into the lab that afternoon. We had a good night's sleep and got woken up at 5:30 the next morning by what sounded like a chicken being eviscerated outside our window, which actually turned out to be something called a wattlebird. Anyway, a few hours later, and without any more sleep, I went into the lab and was introduced to everybody. That was the day of the weekly lab meeting in Marcello's lab. And I can remember it pretty well because I was introduced to everybody in the lab, and somebody was presenting some science, and they started talking about this, and Marcello corrected them and filled out part of the background. At which point, I said, well, actually, I disagree with that. And there was a sharp intake of breath by everybody in the lab. Nobody had done that and got away with that for decades. So Marcello said, well, explain what you mean, very patiently, and I did, and he argued his point, and I argued mine. And 2.5 hours later, the lab meeting finished. And I thoroughly enjoyed myself. It was fantastic to actually have an intellectual companion, somebody who could argue more pointedly than myself, and who could provide so much input. It was just fantastic to me. So this happened at many of our lab meetings after that, until somebody in the lab actually took me to one side afterwards and said, for Christ's sake, Brooke, just shut up in the lab meetings. Well, you don't wind them up. We've got work to do in the afternoon. So after that, we figured out that the way that we should proceed was that the lab meeting would finish on time, and we would repair to Marcello's office, and the discussions would continue. And what I realised was that Marcello would perform sort of academic assault and battery with this huge range of reference that he had, all this data that he had behind him. But he also worked by talking the living daylights out of everything. And these discussions would go on for very long times. This was a war of attrition on the Italian front. So I thought that this was an unfair tactic. So one afternoon, I deliberately cleared the afternoon, had no experiments running, and launched my assault during the lab meeting. We went to his office afterwards. 4.5 hours later, I gave up because I had to go home for dinner. So I realised that wasn't the right way, but I have thoroughly enjoyed our conversations, and I don't think, when both of us have been in Flinders at the same time, that we've missed a single day, because we move over all of the stuff that we agree on and find those little things where we can get stuck into each other. And it's been the most rewarding and interesting and fulfilling experience to have those discussions with Marcello. So that's my tribute, which is about as much as you'll get from a Southern Englishman, I can tell you. So when I came out to Flinders, the project that Marcello wanted me to work on was funded by the, I think it was the Department of Health, to study the effects of opiates on enteric neurons. Now, Marcello was working with Steve Johnson, who had worked in Alan North's lab, where they had worked out the effects of opiates on enteric neurons. And the project in Flinders was to study this and to look at the tolerance and withdrawal mechanisms that actually occur within the enteric nervous system. And the project was going terribly. And it was going terribly for some very simple, pithy little details, which was that when you stick a microelectrode into an enteric ganglion, you encounter lots of the big cells, but very few of the small cells. And it turns out that the opiates actually affect the small cells. So this work was appallingly difficult and very low productivity. People, 2 or 3 setups in the lab could get about 1 cell a week, because you had to hold these little tiny cells on the end of a microelectrode for an hour. It was impossibly difficult. Well, Marcello had gone over to Alan North's lab in 1997, I think, and had learnt a technique that Alan had set up to culture enteric neurons. And what Marcello wanted me to do was to impale these cultured neurons, because that should be a whole lot easier and more productive than this. Well, what Marcello didn't realise was that Jeff Bernstock, another name we've heard a lot of, who was then working in University College, had actually approached me a couple of years before to find out if I'd be interested in going to University College to record from cultured enteric neurons. That was an offer which I found very easy to decline, because I thought at the time that studying the physiological properties of cultured enteric neurons was fractionally less interesting and relevant than studying the electrophysiological properties of the Petri dishes that they were actually being cultured in. So when Marcello told me this, I thought, well, I'm not going to be lasting here for very long. But there was something absolutely wacky about these cultures, which I would describe as basically what you do is you digest a piece of tissue, you pick up the ganglia with a pipette, you put them into a Petri dish, they stick to the bottom of the Petri dish, and then something completely crazy happens. The ganglia explode. They spread themselves out, and the neurons all go out individually to all 4 corners of the Petri dish, that's for the aficionados. And after a few days, they then reaccumulate, and it struck me that this was really weird behaviour. It's the sort of thing that you could possibly sell to the CIA, if you could get brains to explode like that. And perhaps we could use this for recording from human neurons. Anyway, I was kind of interested and intrigued to find out how this worked, but that wasn't part of the remit of what I was supposed to be doing. So I did a series of experiments without telling Marcello or anybody else what I was doing, which involved partly culturing bits of intact gut in plastic Petri dishes. And remarkably, the ganglia didn't explode. And I suddenly figured out it wasn't the culture medium, it wasn't the Petri dishes, wasn't any of this stuff. It must be something to do with actually digesting the tissue. So I told Marcello about this, and he said, oh, you better show me that. So he had a look down the microscope, and sure enough, it all looked pretty good after about 4 days in culture. And Marcello then got very excited. He said, let's do some histochemistry. We had a look at the histochemistry, and sure enough, the tissue was looking remarkably normal, just as if it had been freshly taken out of the animal. And Marcello saw the significance of that. And I can't remember when it was, but at some stage during the discussions, we realised that we had a preparation which we could use for retrograde tracing of enteric neurons, something that hadn't really been done in any sort of systematic fashion, and that this could provide the answer for being able to identify the special little neurons that responded to the opiates. Well, we developed a technique for applying a dye called dye-I that had been ordered by the lab. Actually, the lab had, another one example of serendipity, the lab had actually all bought the wrong form of dye-I. There's 2 forms of this stuff, one with 18 carbons, one with 12 carbons. The 18 carbon stuff doesn't work, and that's what everybody else was using, but they had mistakenly bought the 12 carbon chain stuff, and that worked beautifully, and we could suddenly retrogradely label these motor neurons from the circular muscle and all this sort of stuff, and for the first time see the motor neurons in the ganglia, and we knew that these were the ones that we really wanted to record from. So this turned out to be an incredibly productive approach. It came from serendipity, from a totally stupid experiment, as I look back on it, but we've probably published 30 papers, something like that, on these approaches, including with other colleagues, and particularly with David Watchow, working on human tissue, and this is a slide taken from some work that his PhD student did a few years ago, characterizing human circular muscle motor neurons in the colon. And you can see that this red neuron is immunoreactive for nitric oxide synthase. And Zanmin Song was doing his PhD in our lab at the time and provided a huge amount of energy systematically working our way through different classes of enteric neurons. Well, I can remember going into Marcello's office, and I think it was about early 1994, and during one of our fairly vociferous discussions, saying to Marcello that I reckon that by the end of that year we'd knock off the entire guinea pig small intestine. We'd have all of the classes of cells and would be able to describe it. Now, I trod very firmly on Marcello's toes when I did that, because that was something that he'd been working on for about 10 years, trying to do this using just immunohistochemistry. And I think that really spurred things on, because over the next 18 months, Marcello did a huge number of experiments characterizing all of the classes of enteric neurons using immunohistochemistry, and published a diagram very similar to this in 1996, which John referred to, which is really the benchmark in understanding the types of neurons in the enteric nervous system. And it's still that basic account is still largely correct and relevant. It's had a few minor details have changed in the last few years, but nothing significant. And it's really probably makes the enteric nervous system one of the best characterized parts of the mammalian nervous system, even to this date. So this is a summary of some of those interactions that I've kind of alluded to with a few other people. James Vickers was a PhD student in the lab, and we'll hear from him later on, and Zanmin Song. Steve Johnson and the Alan North connection. Ian and Judy. Jeff Bernstock playing a pivotal role in this network. And as I said, this is just one tiny part of this network, spreading over to Reno, and recently Nick Spencer's come to Flinders and is collaborating with us. From a different connection through Bob Bywater and Molly Holman. It's just a fantastic, huge international family of people who meet up at conferences and generally have good arguments over a glass of Shiraz. So what about the future? Well, again, this comes down to very personal stuff. In 1997, I applied for a research fellowship from the NHMRC and was turned down on the grounds that my research wasn't clear which was my research and which was the stuff that Marcello had driven, and that maybe I was just Marcello's technician. Now, Marcello dealt with that with wisdom and maturity and generosity, because he helped me to set up my own project, give me space within the lab to set up my own project separate from what he was working on. And that's turned into one of the major directions of our lab over the last few years. We started recording from sensory neurons, and we developed this technique that Ian mentioned. A very bright medical student wandered into the lab one day, said that he was a bit bored with medicine and he needed something to keep his brain active. This was a guy called Brady Tassica, and he actually was the one who developed that dye-filling technique. Well, we've applied that to the sensory neurons, and we've found for the first time that we're able to identify the sensory endings of sensory nerves in the gut, and Vladimir Zagorodnyuk has been involved with that, is now doing the same thing in the bladder. And we can start to see these endings, which are really the important sites where these sensory neurons are activated from within the body. Most recently, we've identified the sensory neurons that mediate pain from the gut. And it turns out that their sensory endings are primarily on the blood vessels, and in particular on the blood vessels in the submucosa, which has turned out to be a bit of a surprise, and that paper's now just been accepted for publication. So where is this stuff leading to? Well, we've got a work in progress. We've identified now some of these sensory endings in the gut. These play an important role in triggering pain, a major symptom and a major problem. And those are particularly these endings here in the submucosa. There are other endings in the ganglia. These are the ones in our stomach that tell our brain that we've had enough to eat. There are other ones in the rectum which tell us that this is a really good time to maintain social standing, and it might be a good idea to change the topic of conversation and go to another place. And there are nerve endings outside of the gut which are also involved in pain pathways. But there are many nerve cells that are still missing from this. And what I'd really like to be able to do over the next few years is to work with a great bunch of people. And I've mentioned Vladimir and Penny Lin has done a lot of this work. Marcello and I, working together now for 20 years. Shin Yung Song has left us, but Nan Chen and the other people in the lab are doing terrific work towards this goal of trying to identify all of the sensory nerves so we understand how you get sensations from the gut. And I guess the future, one of the futures anyway, is understanding how many of these sensory neurons there are from the viscera in general and understanding how they work. Because these nociceptors that Ian talked about so much, just in the single cell have remarkable integrative properties. They are outside the blood-brain barrier, and in theory it should be possible to target those with drugs and interfere directly with their function in painful scenarios. So what we want to understand is exactly how they work, what mediators affect them, and how we can manipulate their excitability. So that's what I'm hoping that our lab will deal with over the next few years. That's just a few pithy little details about the sort of research that we're doing. Thanks.
Thanks very much, Simon. Let's just have a short break, no more than 10 minutes, so grab a cup of coffee and please make your way back to your seats. So thank you for hurrying back to your seats. It's my pleasure now to show you a movie from an absent friend. This movie has been couriered all the way from Gothenburg, from Anders Lehman, who apologises that he could not also make it across the ocean. So I'm hoping the sound level is going to be right, and I will just...
Dear Marcello, I deeply regret that I cannot be there in person today to celebrate you, but you can rest assured that I am there in spirit. We have now known each other for 14 years, ever since the signing of the collaborative agreement between Flinders, Royal Adelaide Hospital and Astra. And today we can conclude that that was one of the most successful collaborations that Astra ever has had. My first impression of you, Marcello, as a scientist was that this is a man with a mission. Many other scientists merely scratch on the surface, but you asked important questions and then you went to look for the answers. And in most cases you would find them as well. Being critical is, or at least should be, the hallmark of any scientist, but in many cases criticism is unidirectional towards your colleagues. I don't think that's true in your case. I think you distribute your criticism both to your colleagues and to your own research. Today, we scientists feel compelled to focus only on our own speciality, and perhaps we don't know as much as we should about other fields of science. In your case, that's certainly not true. You have a breadth of knowledge that has always impressed me a lot, not only in bioscience and science in general, but also in subjects such as music, art, politics, and so on. I don't think there is any subject in life that you are not familiar with. I have now visited Australia 21 times, and we have met at most of my visits to Australia in conjunction with the joint scientific group meetings, which have taken us to wonderful places such as Ayers Rock and Palm Cove. And those meetings have always been very stimulating and exciting, and your contribution to the meetings cannot be overestimated. You have an unusual talent in spotting both the merits and the weaknesses in presentations, and you have an ability to force people to think twice about their results and the conclusions. Sometimes it can be quite challenging to discuss with you because of your incisive intellect, but at the end your comments have always been very helpful and very much appreciated by me and my colleagues. I could go on for a long time, Marcello, but I think I should stop here because I know that there are many people lining up to say your praises. Suffice it to say that for me you have always been, and you still are, a role model in addition to being a very dear friend. Marcello Costa and retirement, those words do not mix well together, and I'm sure that the pigeons in the park will be very disappointed. At any rate, Marcello, I wish you the very best for the future, a happy retirement, and I trust that we will see each other soon again. Take care. Bye-bye.
So you see from that video that there is a bit of confusion about whether Marcello is going to retire. I think he's decided to wait until the end of the day, and then he's going to take a vote on those who think he should or those who think... So you have quite a bit of sway. Okay, now, to start off our next session, I have great pleasure in introducing Tony Crandis, who's come all the way from the University of Ottawa. Thanks, Tony. Just do what someone does.
Yeah. I want to, before I say anything, I want to say to Alan North, loss of serendipity, rubbish. Okay? Marcello, everybody's been singing your praises about your contributions to science. That goes without saying. But I want to thank you personally for being an extraordinary teacher, teaching me rules that are very important in science, and passion for the subject that you're studying. And I always teach, and students say to me, why are you so passionate about your teaching? I said, I had a good teacher. And if you are passionate about this subject, it's easy to teach. Thank you. I'm going to tell you my story. It's a little bit different, and I'm just waiting for this to load. I come from the Great White North. I moved there after finishing a PhD at the University of Adelaide. But I started my scientific career really in Marcello's lab when Marcello was my supervisor for my honours degree. I think I can start here. I'm not going to show all these. Okay, can I just hit full screen, that's it? Slideshow? No, down here, there it is. That's it? There we go. Good, thanks.
It's spacebar.
Oh, okay, spacebar, go forward.
But it's still loading up.
Yeah, I'm sorry, these are big images. Okay. So going forward is spacebar?
Yep.
Okay.
It's still probably loading.
Canada's a lovely place, as you can see, particularly Quebec. That's where my cottage is. And that was a few days ago. I think it's all loaded. It's all loaded.
Should be going.
That's what I'm saying. Should be there. Okay. So at the defence of my honours thesis, at that defence was John Finesse and Marcello Costa. And one of the questions they asked me was, could you explain where you purchased John's saline that you used in producing the Krebs for your organ bath preparations? And of course I proceeded to explain in great depth where I purchased John's saline and the constituents of John's saline. But in fact it was John's saline. And everything in John's lab migrated to Marcello's lab and vice versa. I hit spacebar and nothing's happening. Oh, here we go. Sorry, how do I go back now? There we go. And so my honours thesis, which was in 1977, was my introduction to the enteric nervous system. I wanted to do work on CNS, but in fact went down, met Marcello, and he says, well, you should look at the enteric nervous system. He said, what are you interested in? And I said, I'm interested in GABA. He said, well, there were some studies ranging from 1958 to 1972, really didn't find anything, but hell, you should look at all the amino acids. So in fact, I proceeded to screen all the naturally occurring amino acids, and I left GABA to last, thinking there was nothing in the story. And in fact, what we found was the very last amino acid that we tested, which was GABA, in fact showed some phenomenal results, and indeed the pharmacology subsequently showed that GABA stimulated both cholinergic motor neurons and also the NANK inhibitory motor neurons simultaneously, and depends on which region you were and how much tone you had, or whether you could see both responses. And this work was done originally in the guinea pig organ bath. And in fact, it turns out that GABA in the gut, contrary to being the primary inhibitory transmitter in the brain, was in fact excitatory. And it still had all the hallmarks of the typical GABAergic system, chloride dependent, all the subunit GABA-A receptor qualities to it. But this was not unusual. Indeed, if you looked at literature, GABA was excitatory in other places, including, in fact, dorsal root ganglia. Okay, so we went on and then subsequently mapped the system, sorry about that, and showed in fact that this was not just a pharmacological phenomenon, that indeed that there were GABAergic neurons present in the gut wall, and in fact they were releasing neurotransmitters and they were targeting interneurons to induce behavioural responses within circuits, and these are evoked responses. And in fact, if you looked at we did the immunohistochemistry, but in fact the original description that we had for GABAergic nerves in the gut was in fact the autoradiography shown in the middle panel, which we did in the guinea pig. The subsequent work, which is on either side, showing in fact autoradiography for the rat and immunohistochemistry for the rat. And one of the intriguing things also was not only were there GABAergic neurons, but in fact there were peri and paravascular GABAergic neurons present in the gut wall. And of course we went on and characterised them using the approach that's well established in the Costa Finesse labs in determining some of the neurochemical characteristics, and where to look at the families of GABAergic neurons based on their co-localisation of other established transmitters in the gut wall. And in fact 3 to 8% of the gut neurons are GABAergic. And in fact, if you look at the distribution of GABA-A receptors throughout the gut wall, you'll see that in fact they're primarily localised on neurons. And here we're looking at, in fact, the GABA-A receptor subunits, and in fact we're looking at alpha-1s, alpha-3s. We did all the subunit description, and in fact, but in addition to the neurons, there were also GABA-A receptors on cells in the mucosa. It turns out they are in fact macrophages. And indeed, the bottom right-hand image is just taken from the literature, showing you that's a typical macrophage distribution, and the one with the mucosa shows the localisation of alpha-3 subunits. So it's a vast system. And in fact, we did apologise again, and all electrophysiology has been extensive. Our own work has in fact gone on to characterise that fully about 50% of the myenteric neurons have a GABA response, but it's prominent on the AH neurons. In fact, the AH neurons have a slow inward rectification. And noise analysis in fact shows that the alpha receptor subunit in fact is the predominant one, and it's important for those sort of long opening times. That's important because if you look at all the subunits, and we went right through from that constitute the GABA A receptors, it's the alpha 1, alpha 3 and alpha 5s together that are present that we localise on neurons. So it's real, the GABA A receptors are present. And 50% of all the cultured myenteric neurons express these subunits. But in fact, the GABAergic systems only is 3 systems. So the work that I did with Marcello was centred on the GABA A system. We subsequently moved then to the GABA B system. There is a C system as well. It's more recently characterised, or beginning to be characterised. We're only as good as our tools, and the C system is a bit slow in being characterised. But the question then is, what about the B GABAergics and what about the C GABAergics? Okay, and in fact I'll point out at the top right-hand page there, you should listen to your supervisor. In this case, this was David Kerr, this is a review by Graham Johnson, 1978 review, which says, and he wrote on there, beta-chlorophenyl GABA, we should test it in the gut. And in fact, I said, I was too busy. And subsequently, the UK group, Norm Barry's group, made the characterisation of baclofen in the gut. But in fact, the B GABAergic system in the gut is actually inhibitory. That is, if you stimulate the B receptors, you actually inhibit the cholinergic responses. Here characterised, in fact, where we set up a cholinergic twitch response, you apply GABA, you see a stimulated response, but also an inhibition of the cholinergic twitch. You put in baclofen, you see exactly the same thing, and the baclofen response not touched by bicuculin. But the bottom line here is that we come down to the cartoon on the right at the bottom, where you see the cholinergic motor neuron and the NANK inhibitory motor neurons. There are subpopulations, of course, but the A receptors are targeting the motor neurons at level of cell soma, but the B GABAergic system is prejunctional, along with the whole plethora of I call them volume controls coming in on the cholinergic motor neurons. So this is in fact the GABAergic system in the gut. Please bear with me. So we characterised the whole system. We were able to develop a cartoon of the system, showing you that in fact it's in all regions. I apologise. I don't know what's going on. And in fact, a lot of the techniques we've applied here are the typical techniques that are in fact set up in Marcello's lab. We then subsequently went out doing my PhD, and in subsequent years been applying a combination of immunohistochemistry, localization and organ bath work, but we moved that forward then to in vivo whole animal work. And in fact, that's characterised along the bottom, where a lot of our work had been concentrating most recently, and we are getting very similar results as matching up, of course, with the in vitro work, where we're showing that, in fact, the GABAergic system is a very profound controlling system within the rat, guinea pig intestine, and now in the pig intestine. But more than that, it's not only targeting motor responses, but it's also targeting and stimulating the release of a variety of factors from a variety of different mucosal cells, and also at the level of the mucosa where it's localised in endocrine cells. So this is a vast system. And all this came about, this characterisation, from the initial seed, where during the honours program, there was a throwaway note, come on, do an honours degree, examine the amino acids in my lab. So the question is, where's it going? Well, I've always thought that the story in the gut was only half being characterised initially, because everyone was looking at the cholinergic motor neurons, but then you had the inhibitory side, and in fact the cholinergics are constant across all the gut, across all species. But what's different is the inhibitory story, where the inhibitory motor innervation changes region to region and across species. So I think that was the most important thing as far as I was concerned. And we started mapping then what GABAergic neurons were targeting what inhibitory inputs. And primarily GABA is targeting the nitrogics, at least in the proximal intestine and in the antrum. We were able to then expand also by the story and investigate further by doing in vivo recordings, which is the basis of most of what we've been doing recently. So what's this leading to? Why are we looking at this system? I guess the question you have to pose is the other way around, is saying, well, is the system going to allow us or give us target sites for developing therapies for the ongoing pathologies? That's what it's all about. I mean, that's what we're doing this for. We're trying to determine what are the circuits, what are the control mechanisms associated with normal health in the gut, and subsequently then look at disorders. Well, the GABA-A story is, I think, a dumb way to go, and that's been proven because of the fact that the A receptor system is such a profound system in the brain, and subsequently if you develop therapies for that, you've got too many side effects. A lot of effort was put in by drug companies for the big GABAergic story, for baclofen, trying to look at maybe LES-related disorders. But as I understand it now, I think Astra and Novartis have pretty much abandoned their GABA-B programs. So is there a future for the GABA-B targeted drugs? I don't know. I think perhaps with the GABA-C story, we're just now unraveling this. Fortunately, we've got great pharmacopoeia coming out of Tokris. We're only as good as our tools, and in fact, there may be some potential targets for pathology, like the GABA-C receptors, particularly since now that they're getting some papers coming out now looking at the potential for GABA-C in affecting motility. Our own work then recently also gone looking at giant contractions. We're looking at mass movements of colonic contents, and we're looking at how the GABAergic system fits in. The point of this cartoon is that we're trying to find out how the GABAergics fit into the local circuits controlling a specific kind of motor behaviour. And what you see here is that the GABAergics coming in at the level of the soma through the A receptors is tonic. The GABA receptors coming in, input coming in through the B receptors is not tonic. And the question is, how does this fit in? How does this fit into the control of nerve circuits? What's happening? Okay, sorry, there we go. So, and that brings us now to the notion of cultured myenteric neurons. And when you culture myenteric neurons, particularly those showing this low inward rectification, and in fact, and you depolarize them, they start to oscillate. And these are the same neurons, of course, that are being targeted by the GABAergic system. So what we think the future is for the GABAergic system, it may give us a better understanding and may help us look at network control systems. And so we have to look at that in terms of isolated neurons, cultured neurons as a model. And in fact, the Reno Nevada group has been saying all along that the GABA sets the excitability of the enteric nerve control. And of course, now the CNS people are becoming interested in the gut model because many GABAergic people in the CNS are looking at neural network control systems. So now they're looking towards the GABAergic system in the gut as being a model for looking at disorders associated with CNS. So in order to get there, I've been doing whole animal work, of course isolated tissue work, and now isolating neurons for doing our culture work. And cultured myenteric neurons are great. We did this for many years, but the trouble is we were wasting too much tissue. I'm talking about serendipity now. This is a serendipity. So we were doing all this work, but we had an unfunded project and we had students working on this. So how can we save neurons? How can we address the issue of animal waste, controlling our timetable, transporting neurons between labs? We were collaborating with other laboratories across town, with Shell Fuchsa in the Karolinska, and developing neuron banks. Well, we tried putting them in the fridge, didn't work. You can do it with human neurons, but not animal neurons. So we said, can we cryopreserve them and then freeze them? Took us about 4 years, and on the left, you'll see here rat myenteric neurons that we finally these are adult myenteric neurons, which we're able to cryopreserve, which are then, you can thaw them out and culture them. And we've done all the tests against the fresh, and they are absolutely indistinguishable. So we said, okay, that's interesting, we can cryopreserve neurons. So then 6 weeks later, we cryopreserve CNS neurons. So why am I telling you that? Because that led to my forming a company. That's serendipity for you. So now we're selling these neurons. We're not selling gut neurons because there's no market for gut neurons, but there's a tremendous market for central nervous system neurons. And now we've done the technology and taken it to cardiac cells and osteoblasts. And so I started off in Marcello's lab screening amino acids unravelling some of the system, going on for PhD. And part of the models that they applied were from organ bath, whole animal, isolated neurons to determine something about the oscillatory pathways. And now I'm running a company that sells frozen neurons. Serendipity. Thank you.
So I'd like to introduce now Jim Tooley from Flinders. You've only got a 567 kb file. That's wonderful. On this last minute.
Wasn't prepared on the Mac, that was the difference. They should talk to each other, shouldn't they? Mm-hmm. Thank you.
Okay.
Thanks, Judy. Firstly, let me say thank you to Judy for really organising this and allowing me to speak today. It's a great pleasure to have the opportunity as a clinician to speak on this occasion. I feel a little bit overwhelmed because there's been all this neuroscience that's been talked about, and I really don't feel that as a clinician I could really compete. Not that this is a competition. It is going to be a personal view, as I guess they all are. Marcello and I, what do we share? Well, we share a number of things. I mean, we're both of, I guess, of European background, although I seem to have lost my European accent that Marcello has managed to sustain all these years. We're both immigrants to Adelaide, and I guess inspired initially by the Gus Frankel experiment, which was Flinders, and that was the putting together of scientists and clinicians together in the one building in the hope that we might talk to each other, which I think certainly Marcello and I have done over the years. We also had our personal chairs actually awarded at the same time. And in fact, I think Marcello mentioned John Chalmers, who was instrumental at that time for both of us getting our personal chairs at the university. And in fact, I think we even shared a common benefactor, didn't we, Marcello, that we didn't really wake up to until some years later. And of course, I think we're both firm believers in the multidisciplinary collaboration, which I'm going to touch upon. Now, this is one of the things that I guess attracted me to Flinders, and a bit like Bill Blessing, it was the Dr. Costa Finesse situation for me as well. The famous person was someone that I actually, in another city, who wondered why a surgeon wanted to get involved in laboratory studies and wondered whether, in fact, it wasn't as a dilettante or wasting time instead of operating, and possibly even resource wasting. And I think this was sort of the negative, if you like. The positive being when I met Marcello and John at the same time, because certainly initially I had the same problem as Bill Blessing had in wondering who was who. But really both of them, and I think this is probably Marcello that says it, let's talk about it. It's a challenge, whatever it is that you want to do, and let's see if we can make it work. And as a clinician, I guess I was asking the clinical questions that I was hoping that these clever people in the lab would be able to help us try and understand them at least. Even though I think as a surgeon I thought I had most of the solutions anyway. But it was good to understand why the things that I was solving were working. This collaboration has led to a whole bunch of people from surgery that have worked with Marcello and with John at the time when they were both there, and then subsequently with Marcello. And I've just got a list of a whole lot of people whose names are familiar to some of you, a whole host of fellows from a variety of different parts of the world that have worked in collaboration. And I only realised just overnight, of course, that I apologise, David, but I've actually omitted some of the people that have worked primarily with you through this connection as well. And so it just goes to show that really the collaboration has been a very fruitful one for us, at least. And this collaboration has been clearly scientific dialogue. Actually, David, myself, and the other surgeons that I've worked with Marcello, we've worked out how to limit the conversations, because we are surgeons, we're obviously very busy clinically, and he's only got 5 minutes, and that's it. And he knows he's got to summarise it on very quickly, because our attention span is very short anyway. So that's one way of doing it. He's had lots of practical suggestions of how we might do things. He's clearly had hands-on supervision with a number of the people that have worked with us, and of course his encouragement is always there. Now, in the past, these are some of the things that we've done. My own area of primary research, along with Gino Sacconi, who's in the room, has been on a tiny little muscle called the sphincter of Oddi, and we sort of had clinical evidence that it was doing something, that it was causing a clinical problem which we call sphincter of Oddi dysfunction, but we really didn't understand it. And so it was this collaboration which has helped us to unravel the anatomy, to study its motility, some of the neural circuitry, but we still actually don't know why it misbehaves when it does, and I hope that that's sort of ahead of us. David Wojciechow's work primarily has been on the colon, and the work that they've done in collaboration with the neural circuitry, both normal and now verging into the abnormal, I think has made enormous contributions to our understanding of that area. But I know there's a long way to go still, particularly with the pathological problems. More lately, we've been working on things to do with obesity, and in particular, we were interested in how some of the operations that are done around the stomach work. And one of our fellows actually did work with Marcello and Simon, and they looked at least started to look at why some of the restrictive procedures that we use in bariatric surgery might work. That's work unfinished, and hopefully one for the future. The thing that I really wanted to talk about in that area is the second bit, which is to do with the vagus nerve that's already been mentioned by Simon. And as I indicated also, David, and I won't really impinge on his area, is continuing work with the abnormal circuitry of the large bowel. But the vagus nerve and its role in obesity is somewhat serendipitous, and this word keeps coming up actually, and I guess I don't apologise for it, because a lot of clinical observations are serendipitous. And there was a time when we used to actually divide the vagi in order to treat peptic ulcer disease. And this was back in the early 1970s and maybe into the 1980s. And really that surgery was progressing very well until someone found a drug that really made it all redundant, and there was a beautiful operation that went begging. But anyway, if one goes back to that literature, one would note that people that had their vagus nerve divided actually lost weight. And it was that observation that led some discussions which happened between some of us that were interested in this area and a group in Minneapolis, which then suggested whether we might, if we block the nerve, that it might be a way of influencing weight control. What I failed to mention was that in those patients where the vagus nerve was divided, some years later the weight came back on again, because as the neural system would have it, it does repair itself in time, and so those effects seem to be negated in 2 or 3 years after the event. So this is the reason why we thought, well, maybe blocking it may be better. Blocking it intermittently might indeed be better than dividing the nerve. So along with, I need to go back. A company called Enterometics developed this blocking system, which shows at the bottom 2 electrodes and a neural regulator, and these are things that can be implanted and in order to test this experiment in obese individuals. The theory is illustrated here, that you implant electrodes on the anterior and posterior vagi, which is something one can do with laparoscopic surgery. And the electrodes are connected to a neural regulator, which is implanted subcutaneously, and then a message can be transmitted to this neural regulator to create a blocking current on the vagi. The theory, as it started, and I don't believe it's what, as we go forward, this theory may change, was that by blocking the nerve, the vega, what we were probably doing was reducing the motility of the stomach, so consequently producing satiety. We were probably also turning off pancreatic exocrine secretion, which might influence absorption, and this is largely how we thought this technology might work. This is what it looks like. This is the electrode. It's around the posterior vagus nerve. The esophagus is, I don't think there is a pointer. This is the nerve, the esophagus, the head's up this way, stomach in this direction, just to orientate you. This is the liver up the top there. So the surgery is readily doable. And these were the results of what happens to the weight in the first 12 patients that were done. The first 12 patients went to 12 months. We've now done over 50 of these patients. And you'll see that the excess weight loss, which is the way that we measure weight loss in obese individuals, you'll see that it progressively decreases. So by intermittently blocking the vagi, these patients lose weight. These are patients that were obese. They had on average a BMI of around 42, which is a significant level of obesity. When we looked at the caloric intake of a subgroup of these individuals, we were hoping that because of the theory, or our hypothesis was that there might be a difference in what sort of components of the diet might have decreased. We might have predicted that fat may in fact have decreased relatively more than other components, but in fact it didn't. In fact, everything seemed to decrease, and there was no significant difference in the relative differences. But in fact, of course, the total caloric intake decreases over time. And what's interesting when you ask patients how long, for instance, this is to look at satiety, how long, considering yesterday, how long did it take you to feel totally full after starting your principal meal? You'll see that this is how the patients reported it. There is a significant decrease in satiety, and in addition, there is a significant decrease in hunger. This is the feeling of wanting to eat between meals, and these are significant findings. So with this, and this is where we're at, but we're now going back to the laboratory because we want to understand this. We're aware that we're blocking the vagus nerve, 80%, or we're applying a current to the vagus nerve. 80% of the vagus nerve is afferent, not efferent, and yet our hypothesis was on efferent potential mechanisms. And those other nerves, we don't know what we're doing to those other nerves. So here is a clinical question, you see, that we need to go back. And we're fortunate in that with Simon Brooks and Gino And with Marcello sort of barracking in the background, we've attracted some funding now to go back to the laboratory and look at the mechanisms. Having worked out clinically that we do have something that works, we now got to go back and understand how it is working. I suspect that a lot of those afferent neurons are doing something in all of this, but we've got to work out what that something is. Interestingly, when we have actually looked at gastric emptying, the hypothesis doesn't hold up. Gastric emptying is not affected by what we're doing. So that sort of hypothesis has gone, in the humans at least. And the effect on the pancreatic secretion is very minimal also, and we don't believe that that's the way it's working. However, working it is. So our journeys with Marcello as we go forward, well, there have been journeys of discovery. The clinician-scientist mix, I think, has certainly benefited us, and I believe it has worked extremely well for us. We've been taught, we've been inspired, and I think we've inspired a generation of future surgeon scientists. And thank you for that, Marcello.
Thank you. It's now my pleasure to introduce John Dent from the Royal Adelaide Hospital.
Not to make savvy.
It's either spacebar or arrows. Okay. Was your first one a blank?
Yes.
Okay. Thanks.
Well, thank you. It's wonderful to have the opportunity to be here and to see faces from the past, many faces from the past, and to take part in this celebration of Marcello's career. I was asked to talk about gastroesophageal reflux disease, and that might seem rather a far call from Marcello's primary interests. But we really come down to the neuroscience behind inhibition of the mechanical event that is the dominant cause of acid reflux into the esophagus. And this is a scientifically intriguing question to address, but also it's medically extremely important because the prevalence of gastroesophageal reflux disease, manifest usually as heartburn, is extremely high, and in fact it's probably has a higher prevalence worldwide than obesity, with between 4% of Chinese population experiencing troublesome heartburn, rising to 20% of the American population. And reflux disease has therefore become a very major focus for pharmacotherapy. And in the past, when PPIs, proton pump inhibitors, were relatively expensive, it was one of the most expensive or the greatest expenditure of any drug in healthcare systems. So it's a huge market worldwide to find a therapy that is effective against reflux disease. Proton pump inhibitors inhibit the secretion of acid, and they render what reflux is relatively bland to the esophagus. And that has proved to be an extremely effective therapy. But they don't stop occurrence of reflux. And so an alternative to treating reflux disease is to target the occurrence of reflux and prevent it. And the concept as to how acid reflux occurs in excess in the 90s was that the lower esophageal sphincter, which is well established as the major anti-reflux mechanism, was weak because of inadequate signals to it or poor hormonal response. And so this dominated the drug development concept for agents that were targeting the defect of gastroesophageal competence. The problem with this was it was armchair physiology. It was based on an incorrect theory as to what the sphincter was doing at the time of reflux. There were actually no measurements of what was happening when a reflux episode occurred. And we knew by measuring pH continuously in the esophagus that this was very typically and almost universally an intermittent event, suggesting intermittent dysfunction. And so what was needed was measurement of sphincter activity actually at the time of reflux. And whilst I was doing my PhD in Melbourne at Prince Henry's Hospital, and I learned about Marcello and John, and I read their papers, and I was very interested in the control of the lower esophageal sphincter, and that was my PhD topic. I'm sorry the panels come out here, but problems with translation to Mac, I think. What I found in 1978, whilst doing postdoctoral research in the States, using a device that enabled measurement of sphincter pressure continuously, was that acid reflux occurred during this very distinctive, abrupt relaxation of the lower esophageal sphincter. That has not come out, but hopefully the next one will. This shows this event with other channels, and the characteristics of this event was that it was a relaxation that was not triggered by swallowing. When we swallow, we relax our sphincter so food can pass readily into the stomach. Those are the swallows recorded there from the pharynx as pressure waves. And this relaxation lasted about 15 seconds with a very great variation, which is far longer than swallow-induced relaxation. So this has a distinctive patterning which makes it clear that it is an event unrelated to swallowing. And in the course of gathering data on the relative importance of this at Flinders, when I went back there in 1978 and spent 8 years before going to work at the Royal Adelaide Hospital, it became evident that making these measurements in people with reflux disease, that in around 90% of the reflux disease patient population, transient relaxations accounted for between 85 to 95% of reflux episodes, making this relaxation a key target for drug therapy if you wanted to prevent occurrence of reflux. I don't want to go into the technicalities of how these patient populations are distinguished, but around 10% of patients with reflux disease have a somewhat smaller proportion of reflux episodes attributable to transient relaxations. But they represent only about 10%, those with severe esophagitis or large hiatus hernias. I didn't really discuss this at great length with Marcello and John, though they were down the corridor, but every now and then I did talk about my interest in these transient relaxations, and we talked about pathways. And in the course of this time, I was collaborating with Chris Martin at St Vincent's Hospital in Melbourne, who was very experienced with using dogs. There were data that showed that dogs reflux. So we developed this model whereby we insufflated gas into the stomach, recorded from the sphincter and measured reflux. And we found that we could trigger very large numbers of transient relaxations, which were exactly like those in humans. So this became the main workhorse, and these studies had to be done during wakefulness, so these had to be trained awake dogs, because sleep and anesthesia abolish the occurrence of a transient relaxation. And using this model, we were able to define the major patterns of transmission of information to the brain. We are again talking about vagal signaling and gastric sensing, and to come far enough forward to say we can now start looking for a specific anti-reflux drug effect. The development of the Nerve Gut Research Lab at the Royal Adelaide Hospital was triggered by an inquiry from SmithKline Beecham to me for setting up a lab using Factor F funds, Senator John Button's wonderful scheme. And so I was nudged into developing a basic science laboratory. And that was funded for 3 years by SmithKline Beecham, looking for drugs that would inhibit transient relaxations. When that funding ceased, I approached AstraZeneca, and after a bit of persistence, they became interested in this project, and they said, well, we will set up a collaborative arrangement, but we want you to collaborate with Marcello Costa and his colleagues to form a tripartite collaboration in this project between AstraZeneca in Sweden, Marcello's lab, and our own lab at the Royal Adelaide. And hence Marcello became very involved in this issue, and Anders Lehmann, who was talking on the video clip, attests to Marcello's contribution to this particular project. Just to give you an overview of the mechanical components, there's this prolonged relaxation, but there's also selective inhibition of the diaphragmatic crura, and also a very vigorous longitudinal esophageal body muscle contraction. And these motor components, although we still lack a very sophisticated mechanical analysis, appear to really very actively promote reverse flow. And we know they're controlled and coordinated by the hindbrain. So we come down to the question then of what is the sensing mechanism and can we target it pharmacologically? And here we come to a lovely study done by Vladimir using retrograde tracing, marking the point where sensory signals were recorded as the hotspot. Here's the IGLE. Here's the recording using the von Freyhair stimulation, and that correlates with this sensing mechanism. And IGLEs in the upper stomach are the most potent mechanism for triggering transient relaxations. And we know that a range of drugs can reduce signaling up the vagus, and that these inhibit transient relaxations. So just to give an overview of these controllers, the controller of transient relaxation, vagal signals pass up from IGLE to the hindbrain control centers, and a inhibitory motor discharge, well, it's a combined excitatory and inhibitory discharge, leads to those motor events I mentioned. So these have proved to be the major sites for action for pharmacological inhibition of transient relaxations. And these are the sites that Marcello and colleagues have discussed so much in our joint scientific meetings every 6 months, either in Europe or the States, and the second meeting in Australia. And Ashley Blackshaw and colleagues have developed a means of studying gastric mechanoreceptor activity using standardized stimuli and candidate drugs for mechanoreceptor inhibition. And these are our dynamic Swedish colleagues. And Anders is the one on the right, and he was rather more animated, I'm pleased to say, on the video. And this is travelling around a bush road on the Eyre Peninsula on the day they arrived, and I think I might as well have left them in bed. So they were on the wrong side of the world and the wrong side of the road, and that was a tough reality that we had in this collaboration. Now, Tony Krantis, I'm pleased to say that the GABA-B receptor agonist program is alive and well. This shows the discovery of baclofen as an inhibitor of transient relaxations, showing a very potent inhibition, and yes, at the price of side effects, but consistently in the 3 species that we were able to do these studies in. there's been a huge effort to synthesize novel GABA-B agonists which are free of CNS side effects. This shows the effects on transient relaxations in that dog gastric distension model, which has been used several thousand times at AstraZeneca in Sweden. And baclofen is a very potent inhibitor, but there are visible side effects, whilst AZD3355, which is the lead compound in the current clinical research program, has a much greater therapeutic margin, and you have to go to very high doses to get visible side effects. And there are data that show that this is when the GABA transporter in the brain is saturated, so that the first part of this biphasic curve is attributable to inhibition of gastric mechanoreceptor discharge, and the second part is to effects on hindbrain centres. So to come back to discoveries in the neuroscience of the inhibition of transient relaxations. Marcello, Simon, Ashley Blackshaw, and our colleagues from Sweden have discussed these questions at great length. What we want to achieve is the highest effect level, and can we inhibit gastric mechanoreceptors even further with agents other than 3355? Can we attack the hindbrain pattern generator without producing a burden of CNS side effects that is unacceptable for treatment of a problem such as reflux disease? And what other receptor mechanisms could be targeted? And we have very high levels of effect from mGluR5 antagonists. And there are other lead compounds or other receptor mechanisms that could be playing a role. And Marcello's inputs into this program, as you heard from Anders Lehmann, have been fantastic. And for me, it's been a wonderful experience, Marcello, to see this almost serendipitous discovery of 30 years ago being carried through into wonderful science, insights mechanistically into now a promising drug therapy in the form of 3355 in fairly advanced phase 2 clinical trials for reflux disease, and that involvement and partnership with you, Marcello. It's been great. Thank you. So I'd just like to summarise to say that we do have a convincing mechanism for inhibiting transient relaxations. This has been driven by neuroscience. It has produced a promising compound that is in clinical trials, and this has been a true partnership. But before I close, I do want to recognise my colleagues in the Nerve Gut Research Laboratory. It's been a big combined effort, and more I've been a bemused observer at all of this technical wizardry and wonderful science, but I guess that's the way life goes. And I particularly want to acknowledge Ash Blackshaw, who is the mainspring of this project and others in the laboratory. Thank you.
Thank you, John. It's now my great pleasure to introduce Janet Keast.
While this is loading, thank you to Judy and Ian for organising this and for allowing me to be on the guest list for directly participating in this way. It's really a very fun occasion. I would also, having heard our last very interesting talk, like to commend Judy on her programming, given that John's just told us about fluid movement rostrally through the esophagus, and as many know, I work on other body fluids moving somewhere else in the other direction. So that was quite an interesting contrast. Okay, for setting the scene for those of you that don't know where I fit in, I believe I was the third graduate student in John and Marcello's group at Flinders, and this is just one of the many pictures from around that time. It was an extremely happy PhD time for me. Some PhD students talk about all the trials and tribulations and traumas that they go through, and I really can't think of any. And I thank both of them really for that experience. I should point out that through some technical requirement of Flinders University, I was required to select one supervisor, and John indeed was my primary supervisor, and I'll save all my thanks and accolades for him for another time. But Marcello, even though he was officially my secondary supervisor, played a very major role, I think, in my intellectual growth and my enjoyment of that period. He may think that perhaps my intellectual growth wasn't as quick as it should have been, but I hope I'm getting there in the end, Marcello. This photograph was taken at a day in the courtyard at Flinders Medical Centre. As you can see, Marcello's looking very bright and sparkly there. You can see his aura glowing. And as a transition to the next slide, this is my reminder to actually start talking about some science. This is a picture of a culture neuron, and perhaps we could imagine Marcello as the glowing neuron in the middle for today's purposes. If we were talking about this last night, the neuron with which he's closely interacting may well be Daniela, Andrade, his brother, various people. Today, perhaps we think of that neuron as perhaps being Simon Brooks. Simon, I don't know how you feel about that, given your feelings about cultured neurons. And the rest of us are these very grateful glia, just happy to be in the same environment as Marcello. Now, just a very brief piece of history, and I can't take any credit for the quality of the drawing of the guinea pig. That came from Google. As many of you know, the laboratory, of course, for many years focused on guinea pigs. Some of you may not realise that the reason why many people in that area do still focus on guinea pigs is their fear of being bitten by rats. Isn't that true? No. However, the sturdy, admirable guinea pig has served well for many reasons, and of course, one of those may well be because it reminds Marcello of the capybara in South America. And we have the drawing at the bottom of the very elegant enteric nervous system shown its various layers. And what perhaps some of you don't realise is that there's another use of this diagram. Now, my PhD was focused not so much on muscle control and motility regulating mechanisms, but it was focused very much on the layer you see at the bottom circled, which is the mucosa, and it's controlled by the submucous plexus to regulate absorptive and secretory fluxes across the epithelium. So in fact, for me, the value of this diagram was to show how you can very carefully and precisely peel away all the bits like the myotic plexus, which are absolutely of no interest, completely superfluous. You can get rid of them and form a nice, clean preparation of the mucosa and submucosa. So since that time, I haven't actually worked directly on this area, but I have certainly kept an eye on the advances that Marcello and his team and other groups have made. And as many of you know, even though I have much admiration for the sturdy guinea pig, I now work on the much more sort of elegant forms of rats and mice. And my research really focuses on the urogenital tract, so control of reproductive function, micturition reflex, various aspects of pelvic visceral function that I won't be talking about in much detail today. And what I was trying to do in coming up with an answer to Judy's question that really was setting the scene for these presentations today. I was trying to think of a common thread that draws together the work that I've been doing the last few years so I can then present that to you today. And given that some of the work I do focuses on basic physiology, basic anatomy, some relates to nerve injury, some relates to pain following inflammation, some relates to spinal cord reflexes and visceral afferent circuitry, I thought, what in my area draws these things together? And I came up with this very simple little scenario here where I was thinking about what really structures the questions that I'm asking today. And I think when I'm thinking about neuronal communication, or the conversations, if you like, that neurons are having, in the past, mostly I've focused on how the neurons are communing with their targets. And I think in more recent years, my focus has started to move the other way, and that is how the targets are communicating with the neurons. And while this is by no means an original concept, it's something which has, I think, pervaded a number of areas of my research and others, and have really changed the emphasis and perhaps some of the tools we're using, and perhaps actually put us back a little bit further in some ways, because there is much less information really about these processes of communication in this direction. I think Alan North did a great job at firing up people's emotions a little bit, suggesting that we're now in the state where we can actually just do a whole lot of computer work to sort things out. I'd love that to be the case for understanding how the targets communicate with the neurons. But I think my area is a little bit more like Ian Gibbins was saying, that we're actually still at a bit of an earlier stage to do some of the complex modelling. So this will be a talk the next few minutes about neurotrophic factors, and I realise we have some people such as Robert Rush and Zenfu Zhao in the room who are great experts in this field. So as they say in Iron Chef, I do my best. Gambarimashita. Okay, Graham Campbell stole my thunder a little bit last night, as he always has prone to do. This is the Marcello version of neurotrophic factors, and of course many of us know that Marcello first started putting together pasta in his talks as a way of talking about different transmitters, different vesicles, different peptides. And I think we can do the same thing with neurotrophic factors, although I've been somewhat lazy and I haven't grouped them according to their shapes and according to their families, and that was partly because of fear of getting it wrong. But I think, as you can see, we're at a stage in neurotrophic factors where there's a lot of them out there. It's very easy to just get overwhelmed by the detail and to lose track of some of the main concepts that some of the different studies which are occurring in very different areas are starting to tell us. I also use this image taken from Google to remind me to tell you that the next few slides I'll be indicating to you that neurotrophic factors can do unexpected things. And this is a picture taken from a small cafe in Italy where they've in fact made the curtains out of penne. Strange, but true. Okay, so the two groups of neurotrophic factors that I've been studying on and which I'll use for my examples of some themes that I just want to bring out in the next few minutes. The family on the left, which are the neurotrophin family, NGF and its direct relatives, which are of course most critical in the peripheral nervous system for sympathetic neurons and peptide, that is CGRP, substance P nociceptors. And on the right, the GDNF family, which is most critical for parasympathetic ganglion neurons and the...
An envious task is to try to summarize this incredible spur of intellectual output you heard this morning. And it's going to be impossible, so you'll hear from me a bit of a galloping history with a couple of comments which I'd like to part and to share with you. First of all, let me remind you, those who were not here last night, that we learned only 3 days ago, Peter Steele, our former manager of the School of Medicine, passed away. He was supposed to be sitting among us and everything pointed in that direction. Peter was remarkable because he represented the best of what Flinders was and still is. And it shows really the most important aspect of an institution, that is made of individuals. A single individual can make a difference. We were very lucky at Flinders to have many such individuals. We still have. And that is what makes Flinders unique. You don't need great organizations. People make them. So, cheers to Peter. Now, I'd like to tell you that I've been surprised when I did look at my own publication. I actually publish in peer-reviewed papers with 161 colleagues from around the world. This is the number of papers per colleague. You see on the left there are 2 pics. Not surprisingly, you may know what they are, who they are. But that shows that I came in contact with many, many more people that I do remember, and that's a great shame on my side, but I'm recovering a little bit from everyone. That tells you indeed the names. Not surprisingly, John, you and I published about 120 papers, give and take a few. Simon, since you arrived in the last 20 years, we published also significant numbers, and it's no reflection on you, it's a reflection on me that we are not publishing, or I'm not publishing with you many more, because I think you're beginning to publish on your own, and it's quite appropriately so. All the other ones come in order, not necessarily in the most important, but not surprising, you see here very important people, and this will continue down. Even the people I published with very few papers, they were incredibly influential in my life, and that's why I like to share them with you. I published about 100 to 120 papers, so not how many really, because the first one were in Italian. I didn't count very many of them. Some have been in French. I was compelled to write in French, and I refused to quote them, not because they were in French, but because the only people who read them were people from North and East, West Italy and Southern France. But I published plenty of reviews and a few books, as you know, and I'm still writing things which are slightly now more general. But I want to take you quickly. Before I went back to Italy, in Argentina, I started to use my microscope. I was fascinated by what I call pond microscopy. These are pictures of a little individual organism, and I fell in love with life sciences. When I went back to Turin to do university, to do medicine, I encountered a fairly conservative town, as you remember from some of my colleagues mentioned last night. And of course I went back to my mountains, so you heard enough about that, and you may hear a little bit tonight too. There I was actually a student of Giorgio Gabella, you heard. He's a retired neuroscientist, anatomy from University College. We abandoned Italy at the same time. On the right here is a picture of Rita Levi-Montalcini. Rita was a student, the same as I was, in the same department. And she perhaps is one of the most prominent Italian scientists. She was a Jewish girl that had to hide actually during the fascism in 1941-42, and she discovered, going back to some of the science you heard today, the very first nerve growth factor. She called it the nerve growth factor, NGF. She got the Nobel Prize for that. She's still alive. She's 100. She's blind. She's a member of the Senate in Italy, and she is, thank God, or thank not God, is giving hard time to Berlusconi now. She is one of the pillars of democracy in Italy. So cheers to my very first encounter with a formidable lady, just the first of several. Well, you heard actually James Vickers. James Vickers doesn't know, but my first picture of a neurofilament neuron was done in 1964 with a silver staining of the gut. That's why I abandoned it, because I couldn't see very many neurons. But as you know, that was the motivation of starting to work on neurofilaments and for you to go all the way to Alzheimer. This picture at the bottom is the first fluorescent picture I ever took, and I took with my camera and the microscope. I didn't have anything else, and we developed strange machinery to do interesting experiments. And there are some pictures of I still did in Torino. But the big change came in 1970 when I obtained an invitation from Jeff Bernstock, a young professor in zoology. He invited me point blank with a call, and with that call, a telegram. I went to my girlfriend Daniela and asked her, "Do you want to marry me and go to Australia?" And here we are since. As soon as I arrived in Melbourne, I met John Furness. He was still a PhD student. He had the advantage that he read my papers while I could hardly read English, and so I owe to him not only a lot of my science, but a lot of my English. Zoology was a remarkable place in the 1970s. You can see there some familiar faces. In the very middle you see Graham Campbell. Hasn't changed much. Your tummy hasn't changed much. And you were a formidable intellect, and I learned more from you than you care to admit. And certainly I do remember my learning of pharmacology that I knew absolutely nothing, and you were appalled of my lack of knowledge of physics and pharmacology, but you were very kind to me, so I learned a little bit since. Anyway, at that time we were very wild, and I thought that all the departments in Australian University were as wild as zoology. I cannot tell you just how wild they were. It would not be convenient in a public arena. But they were also incredibly stimulating. My mistake was to assume that indeed every academic organization in Australia was like that. So when I moved to Flinders, I tried to impose or to induce something like this. To some degree, some of us succeeded coming from Melbourne, but nothing like the wilderness of zoology in Melbourne. But in Melbourne, Jeff Bernstock was pushing for the idea that one of the mysterious transmitters in the gas was ATP, adenosine triphosphate, which actually Graham Campbell is the one that thought originally when he was doing his PhD, but Jeff never considered that. And so we began to make some fun. He had this idea of a purinergic hypothesis. Purine means is a chemical name for some of the substances similar to ATP. So I contributed to the debate by creating this, been quoted already, this idea that there are some macronergic nerves with a axonal transport of macaroni and macaron synthesize. There are some small packet and large packet. People argue about sizes of vesicles, and there was some reuptake of macaroni. There was some macaronize that destroys the macaroni, somebody eating it, and a macaron transferase when take them away. So that was all done in fun, but not so fun, because actually Jeff was a bit taken aback by the fact that my theory could be just as good as his one. And you heard about the idea that pasta kept going. I'm delighted that the theory of pasta as being transmitters is well alive. And those of you last night that heard my grandson Orlando talking, he's already in the pasta machine, so he's scientifically well-minded as well. We had a very small lab in Melbourne, with 3 of us. When I moved to Flinders, a lucky move, of course, I was attracted by Laurie. Laurie got me to Adelaide. And then they did not interview me. They had a conversation, and then they made me sign a paper, which I didn't understand what it was until I realized I became a foundation lecturer in physiology. That was a remarkable time. These are some of the people that you may recognize. There is actually Robert Rush on the right. Now, I keep saying to Robert Rush, he's the older member of our school. Not in a physical sense, I'm much older than him, but he was here before anybody I realize now are here with us. And when Robert goes, if he goes before me, I'm afraid I'll be the last one standing, and perhaps I should not switch the lights. Then, of course, John Forness was already there, so we had a fairly large group in the 2 departments, mostly anatomy and physiology. Of course, Bill arrives, and you heard enough about Bill to know that we have an ongoing relation, both intellectually, philosophically, and scientifically. Bill started with thinking that he owed a lot to John and I. Of course, now I believe everybody owes a lot to him, and you heard his marvelous talk today. If you thought that he was actually developing in the 1980s, 90s, in the 2000s, in the 21st century, I believe he's doing his best work ever. Of course, we had another mentor, a very important member, not only Laurie, but also John Chalmers. John Chalmers, also a foundation professor of medicine. He was ebullient. He would come from behind and hit your head before you realize, in a friendly way, and he remains that way. He is one of the harder thinker and broader thinker, and we owe him a lot. So thank you very much. I regard you one of my best mentors and friends. Thank you for coming all the way, by the way. Of course, they're good mates. We were all a tight-knit group of people. Peter Howe, I think, may have been here too, but he's also was working with John. Of course, in your fest brief, we all came, and here is the unbeatable Ian Gibbins that I'm going to mention in a little while. And of course, I remain your good colleague and friend. But just to go back to Ian Gibbins, I should mention this because I should have mentioned at the beginning. Ian and Judy, they came in the 1980s. They are the one that thought about this event. And I dream of this event after they propose it. And I think that what is happening, we are in the middle of it. And I'd like to think that they have been successful in convincing me that it was worthwhile trying to live or relive our lives together. And the unfailing resistance of Ian's imagination and Judy's determination and organization make all this possible. I owe them far more than just this meeting. They have been my best young mentors, together with Simon Brooks. Our lab became a bit larger in Melbourne. You recognize there's a young Pat. Pat Williams is still in the extended group. Now, of course, work with Ian and Judy. But we were young and fairly aggressive in our approach. We thought that having come to Flinders was only worthwhile if we made Flinders the center of the universe. We didn't really think about Adelaide University at all, and I think we were right at the time. The group was very varied. You can see here some people you can recognize. On the right, young John Willoughby. John Willoughby joined also Flinders earlier on as a neurologist, as a neuroscientist, and also I regard him one of my best colleagues and friends. I wish I could range his interest. I'm very interested in what he does, brain waves and almost to the link between physics and metaphysics. I follow with great interest his studies, and I wish I could do more myself. The Centre of Neuroscience was a great imaginative endeavor that started from many people and continued to develop over the years. You can see there are some very familiar faces. Now, a little bit of history of CNS. That shows the numbers, the members over the years. Between 50 and 80 now, the number keep increasing. There's a warning given that actually Alan North is concerned that we all may get into the waste bin, and I may be sitting aside, but I don't want to be aside all by myself, so I like to still to keep the rubbish bin empty and keep going. And but there are problems, and of course the problems are mostly that the number of academics is declining for many, many reasons, but the number of non-academics is increasing, which means that academics left are becoming better and better in attracting people. Now this brings me to the fact that I'm very, very touched by the fact that this morning we have 3 fairly senior colleagues. They are scholars in their own merit, in their own disciplines. Anne Edwards, our former Vice-Chancellor, Roy Goldie, who had to leave early to do his job. He was here before. I think if you're still here, raise your hand, but I think you said that you had to do something to do with your job, which is the head of Faculty of Health, and Lyndon Wind, our Professor of Medicine as well, yes, and our former Dean. And between the 3 of them represent the best of Flinders as well. Incredibly dedicated people, as I said, scholar in their own merit. But I must say that you also reflect the difficulties. That's why I appreciate you being here, the difficulties of linking the senior administration sometimes with the people who are working on the trenches, on the ground, like some of us. And I'd like to think at least that you 3 can actually convey to your colleagues that all that obscure activities of us running around and doing things which escape your attention may have come to some fruition, and if not, we deeply apologize, but we had a hell of a good time. The lab at Flinders grew. You can see here a lot of people who are presently here, thank you, and I won't name all of you, but there are so many of you that I feel embarrassed to have to make choices. But we were very productive. At this time, we argue with people around the world. You can see me arguing, it's not surprising, with Mark Gershon, the proponent of yet another theory of another transmitter, serotonin 5-HT. We were proving him right to some degree, at the same time proving him wrong, which is what I love best. And so we enjoyed that time. In fact, we had this dealing and this collaboration with Alan North, who I met only a few years early in 1973 in Banff, together with a young David Hurst. Now, between these 2 individuals at those times, they're the one that really founded enteric neurophysiology. They were the first one to impale the famous enteric neurons. You heard so much about it. They published in the same year and the year after 2 independent papers which set really the scene for the rest of the existence, and so we owe to them a lot. Now, I envy them because at the time I produced a little paper about intercellular recording of muscles. I felt great. I learned a little bit of electrophysiology, but to me those are the gods because it still remains one of the hardest things ever to record from neurons in the gut or for that matter in any other systems. By the early 80s we were able, John and I, to collect in Adelaide, Flinders, the whole collection of people working on the enteric nervous system, and you recognize there are some very familiar faces which I hope will be appearing in some details. You see there again Aaron North, and the major other opponent, Michael Gershon. You can see here 2 happy faces, Janet Kist and Ida Lumen-Smith, who is here. Thank you also for coming. Incredible persons, I would say incredible women. Of course, you know that I do really prefer women than men, and I don't make any apologies for this. And I was very fortunate that they indeed chose to work with me for a short while in their life, and I'm delighted they still regard me as a good colleague and friend. So I wasn't that bad man after all. You can see here a lot of other people, Graham Campbell there, you can see here Jeff Bernstock, and you can see David Hurst. So and here is also Boy Water, who made all his way from Melbourne. So a lot of people. You see John Furness hiding there. He very seldom hides, by the way, but this time he managed. Well, those times were full of meetings. We were around the world very often. John and I, we went to different meetings, so that's why we kind of interchanged pictures with names, because we met different people. We constituted different societies, VRP, substance peace, nitric oxide, any substance, and we were just following them. was hard but very exciting. You can see here again two well-known picture people. Aaron North there arguing all the time with me and with our colleagues in New York. There again. It was a happy time in our laboratory in the 1980s, and clearly Ian and Judy were the one that make a huge difference. Everything you heard about John and my ideas about chemical coding is really due to the enormous breadth of knowledge from zoology and from the proper quantitation. That's what actually, in my view, they brought to us. John Fornés was a physicist, I was a medico. It had to happen, isn't it? I will not accept it, I'll reject it if you don't. I don't use this stuff. At least it's working. So somebody's playing a game. This are the gremlins or my family, I suspect. And I assume there's nothing wrong except that they wonder where the hell I'm gone. Anyway, Ian and Judy brought quantitation, statistics, and accurate analysis of what John Fornés and I, we're just looking at things and semi-quantifying. Let me also tell you that my interest in medicine disappeared very quickly. When I became a doctor, I realized that my best contribution to medicine was not to become a practicing doctor. So I didn't really practice medicine, mostly because I was not really interested in diseases of people. I wanted to know just how people were, how people worked. That's why my interest in well-being. And I think that is also part of the future of medicine. And I'd like to think that my interest and my impact on that might have some bearing of following interest in how things work and how our brain works for the good things, not just for the bad things. I spent time around the world. I was lucky to be invited in a number of meetings with von Euler. There is the Nobel Prize for discovering noradrenaline as a transmitter. I was in very good company, really quite exciting. Among the very first coexistence of neuronal messengers. So after that they used the word chemical coding. But the big things changed after 1998 when young Simon Brooks arrived. He arrived to Australia just in a similar way as I arrived to Melbourne. The big difference, Simon, was that you could speak English and I couldn't. You still speak English and I can't. Well, we had a great time together, and we still have, and I'm very privileged still to be your senior mentor. Now I regard you as one of my junior mentors and senior, because as you probably know, Simon is my boss. Now, talking about bosses, I was amused because, of course, I don't like bosses, and no better person to recognize that is Anne Edwards. I still remember when we found each other at the dinner party, and I introduced Anne Edwards. Well, Anne is my boss, and Anne uttered quickly and without any hesitation, no, Marcello, nobody is Marcello's boss. And so I recognize that, and I thank you for admitting that, but also for allowing me and as many of us to be independent thinker and quite wild activists in science, hopefully not in any detriment to our institutions, quite the opposite. Now, somehow I forgive you for everything, but for this original picture of the arrangement of the neurons in the gut that we call indeed the chicken sort of factory. They all look like chooks. Now, this reminds me what actually Bill mentioned about the shape of the neurons like onions. I do actually believe there are some similarity between chicken, onions, and neurons. And you'll be surprised that eventually in the philosophy of science, they are coming together. The generating patterns in mathematics are remarkably similar. I wish I would know them. Of course, my two most important junior mentors, because they were junior from me when I first arrived to Australia and when Simon arrived, they are looking at each other. They haven't collaborated because, of course, I stand in between. But it's interesting that I have had the chance to pick both of their brains, very different, very rich, and I really acknowledge that I would not be here in science if it wouldn't be from these two guys. So thank you very much. Marcello Tonini cannot be here. Some of you tonight, if you are tonight, you see a little excerpt of his talking. He goes back a long time from Pavia, and he's still there. Only a few weeks ago I was with him. He's very well. By the way, he has been sick, but he's very well now. You heard about Jem Vicker and Zhang Min Song with this beautiful picture, not only in the gut, but all the way to the brain. And so I'm delighted that indeed people starting from the enteric nervous system may very well escape it, as they should. I was lucky to have good groups over the years. It's too short time. The fantastic relation with our first intake student, a young David Watcher, and here is a senior consultant now, he's here. Thank you for coming. Of course, we publish a lot of things together, talking about neuropathology, neuroenteric pathology. I think your work is one of the most prominent in the world still now, and is still going fast, going well. The laboratory under Simon has thrived and is thriving. When Grant left Australia for Reno, we had a good time, as you notice here. We had other people visiting. Vladimir just arrived. D'Antona was from Italy, so we had a fantastic time all the time. You've seen our good friend and colleague John Dent. I admitted to Jim Tooley I did not have any picture of him, or better, I do have some picture which have escaped me now, when we were all sort of watching some whales jumping in the water, all splashed with water, but I couldn't find the picture. But we share a lot of things, as you heard, with Jim. Jim is still a Flinders, and we have over the years interchanged. He gave us a lot of excellent PhDs from surgery. And so our collaboration with John Dent has been fantastic. I cannot comment on what on earth you were doing there with this cup, but you certainly have been a serious and a smiling guy, and we had this fantastic collaboration with Astra. Here he is at your place in the peninsula on the other side, taking our friends from Sweden there. And the Swedish are very friend with Australia because they are very similar. They are similar surprisingly, because one thing very great differences. But we think alike, and has been a marvelous opportunity to interact with them. You heard some comment directly from some of the people you see here. And that takes me to perhaps some of the colleagues that you heard now from overseas. Here is actually our friend from Reno, Nevada. We created, and John Dent is there, and we created a little group in Varena. And I'll mention a bit this tonight because there we were partially guest of my good friend here, Mario Pigazzini, who came all the way from Lecco, northern near the Lake of Como, near Varenna, just above. And so I managed for once to mix my science with my friendship and my culture, as you hear tonight, some of you. Anyway, the Varenna is to develop teaching means internationally and still going, and likes to think that's been a good thing. Many people influence me. Annika Dahlström, she's here already a mother, if not a grandmother, but when she was young in the 1960s, she was the one that used for the first time the fluorescence method in the brain. And that scared me off from the brain for a little while. She was very good, apart from being very intelligent, very beautiful. Other women in science, of course, great influence. Molly Holman, a good friend of Jeff Bernstock. Molly Holman was very influential in bringing me to Australia. I have a highest respect, and I wish her well. I know she cannot be here. She was invited, and I'd like to think that she could have been here quite happily. Elspeth, you heard what I said of her yesterday, and I repeat it. She is another good example of a superb thinker, a superb woman, a superb person, and a wonderful friend and colleague. And Marie Supernant got married to Alan North. I was the one that was the best man. Three women, completely different lives, different part of the world in a way of culture. Three formidable scientists that proved the very point. We missed more women scientists. My experience is that they, for one reason or another, they see the light, they change the directions in many cases, or somehow or other we don't help them enough, we collectively, to make the best in their careers. But certainly they have not only nothing to envy, I have a suspicion that they see things in greater picture, and this is the bit I'm getting old, I'm getting more interested in. They do that naturally. What other better person than Eddie Bulbring? I was very privileged in Banff at a meeting as I got to her retirement. Eddie Bulbring was a German refugee from Nazis in the late 30s, went to England where she thrived, and she is the one that developed the macroelectrode techniques to the autonomic nervous system. She has been incredibly influential, and probably everything we've done since was really started by her. Suna Rosell is a person from Sweden, a professor of pharmacology at Karolinska, that really is the one that compelled poor John Dent to have us also on board with the Astra, because he became, in fact, involved in AstraZeneca early on, also a very dear colleague. Frank Lebbe, Peter Holzer from Austria, they one that really dealt with the substance P, peptides. So that's when we move into the peptide area very quickly and very effectively. Now, I also spent some time in Beijing in 1993. What an incredible change from what we see today. talking about actually numbers, I'm sure the numbers count. I'm sure having big computers count. And if that is the case, then having more than 1 billion people, potential neuroscientists, will count too. So we should take actually great interest in our Chinese colleagues. I was fortunate to have, and I still have, many of them collaborated with me. They are present in Australia, and I was delighted. Now, this is the problem of science. When you start from a little bit, visualizing, naming the top, bottom left, and you go in different directions with 100 of people, thousands of meetings, and in the end, you don't know where you are. So that is a problem of my life. I move around. You've been impressed by the direction in which some of my work and my colleagues taking them is remarkable. It's also a concern because it means that science grows clearly without any direction. At times when people expect science to have a direction. This is a tension that I cannot resolve because you cannot plan too far ahead, and you cannot actually, however, go forever in an apparent random way. There's nothing random about this, but there's nothing really planned. And that's probably how science is. And it reminds me that that's probably what I've been doing myself, and many of you have been doing this. We might be looking in the wrong place for what we are searching, or we may not be searching the thing that we want to find in the right place. I don't know which way it is. Anna North was curious to know what is the fourth dimension. This is me in fourth dimension. 3 dimension is space, 1 in time. I didn't invent it, we live in it. But the good news is that everything can be expressed in 4 dimension. That's me in 4 dimension, I'm somewhere there. And you were asking before, well, it was Marcello's birthday. Is Marcello retiring? Is Marcello about to die? I hope none of them. And the time will tell. But the good news is that we live in 4 dimension, but my new theory, and I'm serious about this, is that actually the reality is made of 5 dimensions. But this is another story, and we might talk about this in the 4-hour session, the one I tend to have with Simon Brooks still nowadays. A little sad aspect, but still very warming for me. Just as Nicky, a dear colleague, I published with him too. He came from Hungary, and he was in anatomy, and he died many years ago. And we taught anatomy together. Actually, we taught the nervous system medical students. At the end of the year, they had their complaints, and they complained that they could not understand somebody lecturing language. And of course, Charles and I were there. I was convening that group, and we were that scared that they were about to identify 1 or 2 of us. Well, our students actually identify a visiting professor from Scotland. So that was, I felt that I was home. Ronnie Franco, my first PhD, also died, and our dear Neil Della, a very, very top young clinician, having done the PhD also disappeared a few years ago. So life is like this, start and ends. But there are things emerging, and you know my passion for communication and science and bringing people together in a medium that instead of diverging is actually converging. And I deeply feel that the sciences of the brain need to go out now of neuroscience. They need to go into different worlds because those different worlds are all involved in the brain. And I mentioned some of this perhaps tonight. The only thing I can say is that there's a tension between the young and the old. The young are all dedicated to find details. The old, you heard even from this morning, and certainly myself, they like to have concepts. They need to come together. So it's very important that the young mentors the old and the old mentor the young. How? They shall all say that when some sage in the 1800s said, well, when an old person tells you that something is not possible, he or she is likely to be wrong. If an old person tells you that something is likely to be possible, almost certainly will be possible. So this is the bit that the old people can bring to young to encourage them to the future and refrain from saying no, think cannot be done, and the young people should think twice before they enter in a maze which may be infinite, because perhaps there's no plans, and we should accept that. And I don't have any hesitation in saying that this is part of life. I've been very fortunate to be part of this little network of life, huge for me, so huge that it's frightening to see how many people I came in contact with in my life. And this has been one of the most rewarding days of my life, may I say, not only to be reminded what I may or might have done, but to see how many people I seem to have indirectly inspired in a great variety of things that they are doing. And this is probably the reason why it's hard to identify a single discipline or a single method in science. And I'd like to think that this can extend beyond science. We need to know more about the society in which we live, and brain scientists. Even I can be regarded as a brain scientist, Bill bless him, despite the fact I worked too many years in the entire nervous system. It's a bit of the brain that too. And so we have a lot to offer to society, and I encourage you to do so. For me, it has been a great time. Thank you so much, every one of you.